I'm John Hemming. All right. Right. Okay. Well, let's go from the top. Now we've started recording. My name is John Hemming. I have a long track record of lots of different things, being in tech. I've been involved in UK politics, being a member of the City Council in Birmingham and a member of Parliament in the UK. I'm a musician. I've set up tech businesses, and now I'm in my 60s. I'm actually 63. I'll give you why don't I give you a little bit of video as well? There you go. Let's see where we can get that going on the right thing. Yeah, there we go. Hopefully, do we get video, start video? Go on, start. There we go. Hello. Here we go. All right. So I'm 63, and perhaps right about 5 or 6 years ago, I was having a lot of problems with sleep. So I got into like the biohacking sort of world. And I've gone on from there. So let's see if I can share one of my pages. So let's click that, click that. So here we go. So you've got some pictures of me from 2019, 2020, 21, and 2022. And you've got the audio now. And you can see I lost a little bit of weight. All right. So I was a lot of 130 kilos, I've come down to about 85 kilos. And I've been, my background is I had a degree in physics from Magdalen College, Oxford. And so I'm into the science world. And I therefore started studying, what is it that's going on? The basic question, what happens when people get older? I got quite a bit older and I am quite old. And bits stopped working. And gradually bits have started working again, which is quite a good thing. So essentially, I'll go into what I think is the mechanism behind the aging phenotype. And my essential hypothesis is that most of the phenotypes of aging are caused by genomic failure, either in transcription, mainly in transcription, really, or translation. The causes of these are metabolic in nature. Now, you've got the non-coding transcription issues as well, and the metabolic problems that cause difficulties in coding transcription, I think, also cause difficulties in non-coding transcription, which is relevant. Now, I would accept that DNA damage is an issue. That is nuclear DNA damage. I think mitochondrial DNA damage is a much bigger issue. And there are probably other issues caused by entropy, but I think those are minor in comparison to those with the malfunction of the genome. And I think there are a number of tools that can be used to mitigate metabolically driven genomic failure. Now, I'm quite happy to have a conversation about each slide. So if you want to interrupt once I've done a first intro on a slide, then please do. Don't expect me to drone on for. I haven't written it. I've written some slides, but I haven't written what I'm going to say. So if you want to raise points after each slide, please do so. But moving on.
We can ask questions at the end, or yeah, if you have
just ask as you go along. There's no reason. If you get some questions you want to ask part the way through, just ask them. So
okay.
don't wait to the end, because I haven't counted the number of slides. There's quite a few.
Yeah, 23.
Yeah, slide 3 of 23. It does tell you, doesn't it? That's quite good. Anyway, so so we start out. We have a question to ask, which is, how does the cell manage energy? Now, this is different to the body as a whole. So in the body, you've got the thyroid gland, you've got vitamin D. I think vitamin D particularly acts as something which is designed to reduce energy demands when food is less available. So it's an annual cycle, if you see what I mean. But how does the cell cope with varying levels of energy? What availability of ATP when it's got a two-stage process of transcription and translation, both of which consume ATP. So it needs to manage the balance of energy going into transcription and translation to ensure that you've not suddenly got like lots of mRNA transcripts hanging around waiting to be processed by the ribosome when there's actually not enough ATP to go through that process. And similarly, it'd be no good sort of having all the ATP reserved for the ribosome. And obviously, ATP is used for other things. Acetyl-CoA is used for other things. So this is the basic core. How does the cell manage its energy usage? I think the answer to that is by not producing the longer proteins. However, let's go on through this. So transcription, you know, I'm assuming everyone has a reasonably good knowledge of sort of aspects of cellular, you know, metabolism. So you have RNA polymerase 2, and it's there to take the DNA and convert it into mRNA. And we all know it's not all about producing proteins. Sometimes it's about non-coding, sometimes a little bit of cellular logic, which operates purely in mRNA. But so the RNA pol 2 is there to take the DNA when required and create mRNA. And to do that, every roughly 142 base pairs, it needs the histone to be acetylated. And that's quite useful because it's a way in which the process of transcription can be controlled by the availability of acetyl-CoA. So
Wait, so sorry, this might be a basic question, but why does it need this acetylation exactly?
well, this is this issue about how the histone, remember, we're talking about the nuclear DNA here, not the mitochondrial DNA. And the mitochondrial DNA is coiled around a histone, some proteins. Those have an advantage, which is that because it's coiled around the histone, it's harder for it to be damaged. It's protected by the histone effectively. And that's relevant when you look at mitochondrial DNA, which is not around a histone, it's much more readily damaged. And that's an important point. And so what happens is there are a number, not just acetylation, there are a number of there's a number of modifications that can occur to the histone. This is distinct from methylation of the DNA itself. This is acetylation of the histone. And by acetylating the histone, it's opened up, and then this enzyme called RNA polymerase 2 can come down, read the DNA and create mRNA. But for it but for it to do that, the histone needs to be opened up. So the DNA has to be separated from the histone. I'm not sure whether it's actually possible for RNA pol 2 to transcribe the DNA without acetylation. It might be something that happens very unlikely. And one of the interestingly complex things is that as part of RNA pol 2, there is the histone acetyltransferase, and that opens up the DNA. However, it is also possible to have non-enzymatic acetylation as well. And there are circumstances under which that may occur, particularly when the cell's being differentiated. But the main point is that there is this control on transcription, which is that the histone needs to be acetylated, right? And then once it's been acetylated, it can then be deacetylated and closed up again. People talk about SERTs. There are a number of different classes of deacetylation in enzymes. Class 3 are all SERTs, all right? So, but they're only one of a number of mechanisms by which the histone is closed up once the mRNA transcription has been produced, all right? And actually, there's a lot of, when we look at like histone deacetylase inhibition, these are quite commonly known substances like curcumin, quercetin, fisetin. Resveratrol is a HDAC inhibitor as well. There's a lot of them around. And there is a really interesting question that I have, in reading all the literature, I haven't managed to find any indication of what happens. Is what happens once, if you think about it, this enzyme is trundling down the DNA, and it's reading the DNA and creating mRNA. And obviously it doesn't do it instantaneously, it does it quite quickly. And it needs the substrate, it needs the relevant nucleotides for the mRNA, it also needs acetyl-CoA. If it hasn't got those, it can't produce the transcript. So it's going to hang around, and we know from papers from the literature that it gets stalled. And one reason it would stall, of course, is a shortage of acetyl-CoA, because without that, it can't progress. There's a really interesting question, is what happens if it's stalled and a histone deacetylase removes the acetyl group from where it's stored, which we don't really know what happens there. I wonder if that's what creates an alternative splice. I'm sure people are aware of splicing. Yeah, everyone happy with splicing? Yeah.
I'm
Okie dokie.
satisfied with splicing, yes.
Yeah. So there's one of the things about splicing is that's controlled by acetyl CoA availability. So you have this big question of how is splicing controlled by acetyl CoA availability beyond this question of it being necessary to open up the DNA open up the histone so that the DNA can be transcribed. But still, anyway, we know it's stored. So if we go on to the next slide, there are a number of papers, and I gave you links in the previous draft to some of those papers that report that part of the aging phenotype is the failure to produce longer genes. Yeah, there's a number of papers. I had 3 papers that looked at it from different angles. And there are 3 hypotheses as to why the longer genes are not transcribed. There is the DNA damage one. That is, the DNA is damaged, so you can't produce it, so it stops. There is the absence of splicing factors. And the third one, the one I tend to support, is the limit on substrate acetyl-CoA. Now, the reason why I don't think DNA damage is the valid reason for the majority of the longer genes not being produced. I would say it's acceptable at times. I'm sure it does cause some problems. Firstly, if DNA is damaged, then you can't produce it. So it would stop. And what we see actually is a gradual reduction in the longer proteins being produced rather than immediate cessation. And the second one is that point about symmetric cell nuclear transfer. We know that if you transfer a nucleus from an older cell to a younger cell, then it works. It doesn't fail. It works within the lowest nuclear cell. So it's not actually an issue of DNA damage. It's something else about the environment. Now, when it comes to splicing factors, there is quite a lot of evidence in the literature that acetyl-CoA availability affects the splicing decisions, what splices are produced. So, if you have this issue, is it the availability of splicing factors, or is it the availability of acetyl-CoA? Well, acetyl-CoA is known to be part of the issue. And then you have that particular paper on osteoporosis, which I thought was a very good paper, and that got me going on all of this. I think it's and we know that obviously if there isn't acetyl-CoA, then RNA pol2 would stall. I think it's a reasonable case to be put that that's the primary cause of the stalling of RNA pol2. Yeah. So moving on. Cytosolic acetyl-CoA is a key energy availability, and it comes from 2 different sources in the main. One is from conversion from citrate into acetyl-CoA, and the other one is from acetate. Acetate, very often, although you can get it exogenously, you can also get it endogenously from deacetylation of the histone, amongst other things. Now, the acetyl-CoA is used for a number of things. It's used into the mevalonate pathway, which obviously is used for a wide number of things, including creation of cholesterol. It can be used to create lipids, to store energy. It can also be used to create cytosolic ROS, which is important in terms of how the cell responds to other cells in the area. But the key to control on transcription is acetyl-CoA going into the nucleus and being used to acetylate the histone. Now, within the cell, it comes out of the mitochondria via the citrate carrier or the tricomposite carboxylate carrier, which the gene is called SLC25A1. But it can also come from serum. Now, different cells express different citrate transporters in the cell membrane. The 3 main ones that are relevant are SLC13A2, 13A3, and 13A5. 13A5 is particularly expressed in liver cells. And actually, 13A5 is the one also known as INDI, I'm not dead yet, or MINDI, mammalian version of I'm not dead yet. And that is used to actually manage the serum level of citrate. But the important thing about the energy processes is the provision of citrate by the mitochondria to the cytosol is an indication of energy availability. So as there is more energy available at the cellular level, more citrate comes out of the mitochondria. So, an interesting point, and this is one where if you read the literature often is a bit confused. And that's because the level, different cells express different transporters. And one of the things, if we look at the stuff on SLC13A5 dealing with Drosophila, it was found that the less of the SLC13A5 protein that's expressed in liver cells, the longer the flies lived. And the reason for that is that the liver cells draw citrate out of the serum, which means there's less citrate available for the other cells. And there's the progeria stuff. There was another paper which looked at comparing SLC25A1 to SLC13A5. And again, they looked at the liver cell. And the fact is that citrate, to some extent, gets into all of the cells. And it is found that it is pro-longevity to have a greater level of citrate in the serum. The reality is, say, if people get a blood transfusion, they will find they get a massive dose of sodium citrate with us as an anticoagulant. That is generally taken relatively rapidly with a half-life of about 30 minutes out of the serum and burnt up in the liver cells. But a small amount of it goes into the other cells. And in fact, there was an experiment using dietary citrate supplementation, which found it enhanced longevity in Drosophila. And that paper, there's a link there to the paper there. Again, it's another one of the papers I've referred to. So moving on. So going back to the Drosophila paper, the paper about osteoporosis, I looked particularly at what were the inhibitors of SLC25A1 that were also parts of SASP. And the one that is key in this is interleukin 10. And interleukin 10 is part of SASP, and it operates through the Janus kinase, JAK-STAT. It inhibits NF-kappa B, but NF-kappa B is a promoter of SLC25A1. So in other words, as the level of SASP goes up in an area, you get less NF-kappa B, and you, as a consequence, get less citrate carrier, and you get less citrate in the cytosol. And this is particularly relevant when a cell is differentiated, when a stem cell is differentiated. And you see that in the osteoporosis paper. And I think this carries forward to other senescent cells. So my basic hypothesis in terms of senescence is a large number of senescent cells, possibly the majority, we don't know, are cells which are stuck in the process of differentiation because of insufficient acetyl-CoA. And that arises from a shortage of SLC25A1 in the mitochondria. And we know that causes osteoporosis from that particular paper. Interestingly, there was a PhD paper done in Portugal, which found that interleukin 10 can cause senescence. And that's counterintuitive. And the reason it's counterintuitive is that interleukin 10 is actually an anti-inflammatory cytokine rather than inflammatory cytokine, because it's reducing NF-kappa B. But in fact, this particular paper, and when you study the literature, there's not that much looking at what interleukin 10 does. But this particular paper effectively found that it can cause senescence in a number of ways. And we know of the mechanism that would work for that. So what we have, therefore, in terms of translation is that in the cell thinking there is insufficient. Oh no, this is sorry, going back, that's transcription. In translation, there can additionally be a problem, which is insufficient energy there causes proteins, part-created proteins, to be sent to ribosome quality control. Where you see that is when there's a lot of mRNA transcripts hanging around, but haven't successfully managed to transfer into the proteins. I think that's relatively minor in comparison to the failure of transcription. However, moving on. So my conclusion in terms of mechanism is cells need a mechanism to work out how the genome responds to a lower level of cellular energy. And I think that is the system that results in the longer genes not functioning, because basically when it hasn't got the energy, it doesn't produce the longer proteins. And also, I think this also has an effect in the non-coding transcripts as well, and generates the substantial part of the phenotype of aging. So let's look at where this changes. And the EU social Hymenoptera, clonal creatures like bees, are a very good example. So you have the queen bee. And I think people probably know that the reason why the queen bee is different to worker bees is that the queen bee eats royal jelly. Worker bees have it for 3 days, and the queen bee has it for her life. And the queen bee lives for 2 to 3 years. And in the honey season, the worker bees only live 6 weeks. And they have the same genes. So the genome is exactly the same. But because of what the queen bee eats, and it's slightly different with termites. Termites have a queen as well, but the difference is, and I don't think people quite worked out exactly why the termite phenotype is different. But the queen bee lives a lot longer, and it has a different phenotype, or she has a different phenotype. And when you look at it, there's 2 key things I identify in royal jelly. One is vitamin B5, which is the coenzyme I precursor. And the other one is an HDAC inhibitor. Now, it's not a very strong HDAC inhibitor. It operates in the millimolar range. Most of the ones I use operate in the micromolar range. And a lot of those that you use as drugs are actually operating in the nanomolar range. But the important thing is this, is that the combination of a vitamin B5 coenzyme A precursor and a HDAC inhibitor results in the queen bee living a lot longer with a different phenotype to the other bees, the worker bees. So what I've looked at experimenting with on a biohacking basis is interventions on transcription. Exogenous acetate and citrate, particularly the citrate, which produces functional changes. Janus kinase inhibitors are a possibility, and they are used, and they do have the effects you would expect to see. So they have effects on hair growth and things like that. But I don't like interfering with the way in which the cell operates in terms of controlling interleukin 10 and interleukin 6. So I personally am not a fan of Janus kinase inhibitors, but they are used and they have been approved for medical purposes. And the other thing is slowing down a deacetylation, not to stop, but slowing it down. So I use about 14 or 15 different types of HDAC inhibitors. An important other thing to do in terms of dealing with this energy issue is dealing with mitochondrial DNA heteroplasmy. Now, I'll go into this in a little bit of detail. Everyone knows about mitochondria. I think people probably know that the mitochondria have their own DNA when it comes to the electron transport chain. Yeah? Okay, yes. And it's a circular DNA. And they probably also know that this circular DNA doesn't have a histone. Yeah? So it's much, much more vulnerable to oxidative stress. And obviously the electron transport chain generates a lot of reactive oxygen species. So the mitochondria is particularly vulnerable to damage to the mitochondrial DNA from the activities of the mitochondria. And in any one cell, there will be different mitochondrial DNA. And the interesting thing I find is that when the egg cell goes through the process of turning into a zygote, there is a process of a bottleneck to reduce the variation in mitochondrial DNA, which is all inhibited. They kill off the DNA from the father. It's all inherited from the mother, but even the mitochondrial DNA inherited from the mother varies. And obviously, it can vary in such a way as the electron transport chain is not properly produced. So it's an inefficient mitochondria. And the point about mitophagy is that the cell, when it chews up defunct mitochondria, it looks those with the awful membrane potential. Sadly, not all mutations that damage energy production also damage the membrane potential, because then the cell can't identify that. But the important thing is this, is that if we go through making the mitochondria more efficient, then we chop out those cells that are not producing adequate energy, either to feed the citrate carrier and generate acetyl-CoA, or even just to produce ATP. And there are lots of ways of improving mitochondria. Rapamycin is one, because that encourages autophagy. Urolithin A, I think, is the same. Exercise, fasting, calorie reduction, pulsed electromagnetic, I think that works. Red and infrared, AMP kinase activators, ATP enhancers help as well. HIF1 alpha, which I'm happy to talk about at length, very important process. Very interesting how hyperoxia has 3 levels of activity. One is the low level HIF1 alpha, then the NRF2, and then the NF kappa B. Extracellular vesicles, those are also influenced by HIF1 alpha. Plasma donation, again, that's a mechanism for both getting rid of interleukin-10, but also getting some extracellular vesicles. And there are lots of other interventions that improve mitochondria.
What's the relation between caloric restriction and mitochondria?
I think it's actually about limiting the glucose levels, because I think when glucose levels go too high, that creates more ROS. And if you manage your I don't think caloric restriction is brilliant, but I think it is a mechanism for minimizing the. See, the point is this: if you can minimize the damage to your mitochondria, and melatonin is another way of doing, then that in itself is a good way of. There's 2 aspects of this. One is reducing damage. The other one is fixing damage. And obviously, ideally, you want to fix damage. But caloric restriction, I think, is very, very good for reducing damage, which is where I think the benefit of caloric restriction comes from. There is the difficulty of this is this debate of to what extent, in terms of human beings frail in their elderly state.
But then exercise would induce more damage.
Exercise, however, starts HIF1 alpha and gets autophagy going.
Okay.
Yeah. So exercise is one of the routes toward what we want. Caloric restriction is useful, and actually, because it can encourage autophagy, will improve mitochondrial quality. Because mitochondrial quality has to be seen as one of the key elements in terms of improving mitochondrial quality, improving the response to the ATP-ADP gradient, but also ensuring there's an adequate level of cytoplasmic acetyl-CoA in the cytosol. But anyway, so moving on, all right? Yes? Moving on? So I've done a small human trial, mainly me, but there are others, mainly friends and relatives, but there are other people who've joined in on the thing. Now, this has no placebo control or blinding, because people know exactly what they're doing. And as with all scientific experiments, the key question is replicability. And so obviously anyone else can try it out. I've got the XPRIZE team, which will be experimenting with this, and we have some useful results. And I'm going to go in some of the results from me for now, but there are anecdotal stuff is not, in my view, very strong evidence, but it's something that can give people an opportunity to have a look at things in another way. So here's one thing: C-reactive protein. All right, so my C-reactive protein is, in my view, one of the key biomarkers, because what that gives you is an indication of interleukin-6 levels. And interleukin-6 is one of the parts of SASP. Now, when you get an infection, your interleukin-6 will rocket, and then your C-reactive protein will rocket. Okay? But then it will come back down to its average level. So if you measure your blood frequently, you will get a base level of CRP. If you do it once every 3 months, that's not much use at all. I do it weekly, and that will give you an indication of your basic burden of senescent cells. Now, this is one of my measurements, and I don't know how much people are into the biomarkers. It's quite hard to get sensitivity below 0.3 milligrams per liter. But my CRP is normally below the measurable level of 0.15 milligrams per liter. Now, if we compare it to sort of people like Brian Johnson, who I think is 0.2 milligrams per liter, I think that's a material measurement to indicate I've reduced the burden of senescent cells. So that's one thing.
What are we looking at? Is this your results?
These are my results, yes.
Okay.
I just happen to have an image from my blood test here. So my result, these are all my personal results.
There is someone on YouTube. It's called the channel's called Conquer Aging or Die Trying, I believe.
Yes,
And
he doesn't have us. That's Mike Luscard, and his test isn't as sensitive as mine. His may be as good. You know, he can only get it measured down to 0.3 milligrams per liter. I use a number of different labs. The best one for sensitivity measures down to 0.15 milligrams per liter. There's another one that will do 0.16, and there's others that do it to 0.3. Mike can only, he might be better than me. I don't know. I'm not saying he is better than me. I'm not saying that he isn't. All I'm saying his lab is not as sensitive as the most sensitive lab that I use, which is down to 0.15. And mine's below.
No, I mean there might be a lot of interesting biomarkers that there's definitely something there.
There's definitely something there. This is so important because as long as you measure it, because every so often I get infected. So I had COVID about a year ago, and my CRP went up again. Then it came back down again over a period of weeks. And it's a very important biomarker. Another important biomarker is blood glucose. And these are measures from Dexcom. In 2022, before I started all of this, in 2023, over a year ago now, obviously, your blood glucose depends on what you eat. Now, I know I ate the same things on these days because I'm very consistent in what I eat. And if you see, and this is measured in UK measurements, and if you want to transfer it to US measurements, multiply by 18, and you see here that my blood glucose was going above 10 millimoles per liter, and now is below the 0.8, which is about 144 millimoles of US measurements. That's an important threshold because there's a different metabolism. I think it's inositol, but don't hold me to it. There's a different metabolism in the cells that comes in when you start going above 0.8 millimole, 144. I think is it milligrams per deciliter? I don't know what's the US measurement for glucose, serum glucose. I think it's milligrams per deciliter, but it's 140. It multiplied by 80 from the millimoles per liter. So what I've managed to do there is to get my glucose system to respond better to the same challenges. And you can see that it's rather than going above 10, which is 10, above 10 isn't awful. It's just it's improved massively. Cystatin C, creatinine is an awful measurement for kidney function. Cystatins, and the reason creatinine is an awful measurement is it metabolizes in blood. It's very responsive to exercise. If you have more muscle mass, you'll get a greater cystatin C result. Creatinine, these are figures from Sweden, in fact. And on the right-hand side, I've got my measurements. Again, these are some time ago in 2023, but that's because I use this image. And as you can see, kidney function has improved. Now, the kidneys are a key place where you see a lot of development of senescence, which I think is this failure in terms of development. Then we've got in epigenetic age. These are true diagnostic things. Now, on this one, it's only 10 years, but on this one, in theoretically, my extrinsic age is 36. Now, we don't know what the functional meaning of epigenetic tests are, but it's an interesting thing anyway. I personally think that methylation of the DNA, remembering it's methylation of the DNA, is a response to DNA not being transcribed. So in other words, if your DNA is being used, it doesn't get methylated. And there's a sort of balance between methylation of the DNA and acetylation of the histone, where if your transcription is active, it will remove the methylation records, and you will and it will then be more readily transcribed, and it sort of creates a stickiness in transcription where if genes are being frequently transcribed, they're not sticky. But if they're infrequently transcribed, then they're much more sticky. And that's a good mechanism as cellular energy becomes less available to make sure the cell's not trying to transcribe genes that will take up energy unnecessarily. So other things, I have a weekly venipuncture, and you're welcome to see the fact that my arm, I can show you with a macro camera if you want. Let's see whether the camera's there. That I don't have scars on my arm where I have a weekly venipuncture. Slow hair regrowth, well, you know, it's a very slow process. And, you know, you don't know because you don't live in my body, but I've developed muscle mass that I didn't have previously. Obviously, exercise is part of that. And then you've got all these papers. So that gives you a quick run through. I'm very happy to talk about it. I'll take the thingy down.
Okay, yeah. Thanks so much for sharing. Very neat. You also posted, you had some patent or something? What was that
it's only a
about?
patent that relates to all of this, really, which is a patent about using citrate exogenously, combined with other things to do this. It's no different to what I've just been talking about.
It's like a procedure patent, a patent on the procedure.
I can talk about patent law if you want, because I've been spending some time on it, but you can patent producing a substance for a particular purpose. So if you, so if you went and off and bought a load of citrate and you mixed it as I do on my kitchen table to eat it, it wouldn't be covered by the patent. If somebody sells it specifically for the purpose, then they'd have to contribute something towards the research.
That was like, you submitted the patent or it was granted?
It's in the process at the moment. So patent law works, you do a domestic patent. And then after a year and a half, you can turn it to an international application through the Patent Cooperation Treaty. And then that goes, you have arguments with it's the European Patent Office in this instance. So I'm having those arguments at the moment. Whether it will be granted we don't know. You might as well, you know, it's sort of, it doesn't do any harm.
Yeah, John, thank you for the presentation. It's really interesting. Question about rapamycin. Could you please elaborate a bit more how it's related? I think
rapamycin is a mechanism for inhibiting mTOR. If you inhibit mTOR, it is more likely that autophagy will occur. Now, I personally take rapamycin every 3 weeks. And the reason I do that is because I want to improve the quality of my mitochondria. Now, there are debates about how frequently to take rapamycin, whether you should try to avoid inhibiting mTOR2 as well as mTOR1. And I'm very happy to have that discussion now or any other time. But I think if you wish to maintain the efficiency of your cells and function well as a creature, there are a number of things you need to do. Some of them relate to aging, and some relate to cellular function. So when it comes to cellular function, you need things like sufficient copper, sufficient molybdenum, sufficient selenium, sufficient magnesium, various other micronutrients. They all have their different effects. and they all have some help. Some particular things deal with aging, which are, as I said, the autophagy stuff and the acetyl-CoA stuff. And obviously rapamycin's part of that. I don't think you can take one substance and live forever perfectly healthily. And also, let's take osteoporosis. Bones renew every 7 years. If you fix, and it is, I think, accepted, settled science, that the reason osteoporosis occurs is that osteoclasts that chew up the bones continue to be created. Osteoblasts that rebuild the bones stop being created, right? And then the paper says that's because of acetyl-CoA, blah, blah, blah. Now, if you fix that today and you've got osteoporosis, it will take 7 years for your body to rebuild your bones so that you are in a state where your bones are as good as they would have been had you not had osteoporosis. So it's not an immediate process that you suddenly go into a room and suddenly you turn out at the age of 18.
Yeah, and just curious, so I've been taking metformin for the last 5 years. I don't have diabetes or anything, but I'm under the impression that it promotes longevity through healthy gut bacteria.
Yeah,
what do you have any encounters with metformin as well? I'm just curious.
I take berberine. Berberine is a HDAC inhibitor and has similar effects to metformin. I don't take metformin. I don't know if you know, but there is a rapamycin news forum, and we were discussing things on rapamycin news. Now, when I looked at the papers that people were using to justify taking metformin for non-diabetics, there looked to be a selection bias in it. And what it appears to be is that the healthiest category of people were those who were diabetic but did not need to take any medication, including metformin, to manage their diabetes. Now, the metformin category were people who were diabetic and took metformin but did not take anything else. So in other words, all they had to do was take metformin. Now, that excludes those whose diabetes got materially worse. So I do think there is a selection bias problem with that particular analysis. And I don't take metformin, but I'm sure if you find it useful, I'm sure it's useful. I wouldn't say it's not useful to people necessarily. It's just the papers that were being used to justify the taking of metformin were subject, in my view, to a selection bias.
Right, but you're doing berberine for essentially the same longevity.
Well, berberine has similar, but berberine is also a HDAC inhibitor in the micromolar range, around about sort of 20-30 micromolar. So it has that HDAC inhibition function. So you've got berberine, quercetin, fisetin, curcumin, pterostilbene. All right, all of these are HDAC inhibitors, and very interestingly, when you look at historical natural substances that people have taken a lot of historically because they thought they did them some good, a lot of them are HDAC inhibitors, and a lot who are not HDAC inhibitors are AMPK activators as well. And AMP activation is a good thing.
Maybe I'll just ask, so you're saying that the body, when the young body, it does a good job at maintaining this balance of transcription and translation and maintaining an availability of acetyl-CoA and perhaps other substrates.
Yeah, particularly acetyl-CoA, but yes.
And then what happens?
What happens as people get older, their mitochondria deteriorate, so the energy levels go down.
Their mitochondria deteriorates. You mean the actual mitochondrial DNA?
Well, it's because obviously the mitochondria, the electron transport chain, is maintained by the mitochondrial DNA. So if you damage the electron transport chain, then the mitochondria will recreate it in the pattern of the mitochondrial DNA. So the mitochondria deteriorate because damage is done to the mitochondrial DNA, and therefore the mitochondria become less efficient. This is where heteroplasmy comes in, because you get mitochondria in the cell. Because you just go through this again. The mitochondria, theoretically, a bacterium that came into the cell where there was a sort of symbiotic relationship many billions of years ago, right? Most of the genes to create the mitochondria are stored in the nucleus. But some of the genes that create the mitochondria, particularly the electron transport chain, are stored in the mitochondria, the mitochondrial DNA, and they are inherited from the mother. They're not inherited from the mixture of the mother and father. And they are not coiled around a histone. They are very vulnerable to damage. And they do get damaged. And you develop.
It's hard to say what is damage. You could certainly find evidence that they change over time. You know, the egg cells, babies are born young. So
Yeah,
why aren't the egg cells damaged? Why isn't their mitochondrial DNA damaged?
Because of the bottleneck. And what happens is and basically what happens is because of the bottleneck, sometimes babies inherit mitochondrial defects. What it does is it reduces the range of mitochondrial DNA.
Wait, say it again. Sorry, the bottleneck of you're saying when the mitochondria of the father is destroyed?
No, the bottleneck is when at a point, I am not personally 100% exactly when the point is on the creation of the cell or when the egg cell or when the egg cell is fertilized. But at a point, there is a bottleneck that prevents too wide a range of mitochondrial DNA going through it. So what happens is, if you've got really awful mitochondrial DNA, then the zygote will die off at an early stage. But on the other hand, if you haven't, then the range of mitochondrial DNA is lesser. Now, I don't mind hunting up references for this. It's settled science. It's not anything particularly new. I think it may not have had enough attention because it's clearly the mechanism that affects quality of mitochondria, because mitochondria will maintain themselves. As you damage the electron transport chain, it will repair through the mitochondrial DNA.
Are you familiar? We read a little bit of Nick Lane in this group. It is amazing when you read about it, the fact that there is machinery to actively destroy a lot of the mitochondria in the egg cell. It must be important. Yeah, must be doing something.
That's the bottleneck. That's the bottleneck. It's the reason that I didn't know it was Nick Lane, but that's the bottleneck. Basically, they reduce the cell reduces the number of mitochondria. The effect of which is, very simply, the cell has less heteroplasmy. So it has a lower range of mitochondria. There still has some variety in terms of mitochondrial DNA. Now, if that egg cell has mitochondria that are too inefficient, then it won't survive to being born if it's a mammal, right? It will die off. It might survive with mitochondrial defects, which is why you find, even though people inherit their DNA from their mother, a child can have a mitochondrial defect that the mother doesn't have, because the mitochondrial DNA that are inherited after the bottleneck are insufficiently efficient for the soma to function. It's a really interesting thing.
The mitochondria, for all these networks, they're sending DNA. There's certainly changes to the DNA over time. It's very hard to say, if something's damaged. You could just view it as DNA is responding to its environment.
Yeah, so the question is, it's changed, and therefore your question is, does it help? Where do we get to?
There's some change, and it's a very small amount. So I don't know.
Well, it's a question of where you get to with all of this. How again, I don't mind. Look, I posted a summary of what I was going to say on the forum so that you could identify things if you wanted me to get the papers. Now, if you want me to, we could have a further discussion some other week about, and it would be a good discussion to have, where I'm quite happy to do some further research on the literature as to exactly what's happening in terms of levels of heteroplasmy. I think it's quite clear that heteroplasmy is not a good thing. And it's quite clear that if your mitochondria are inefficient, you don't work well. And obviously, it varies from cell to cell. And you have this question, this is the Seafried thing on cancer, that as people move away from using mitochondria for energy towards using fermentation for energy, you get cancer. Right? And that's an interesting thing in itself. So you look at mitochondrial DNA, mitochondrial damage, and I do think the key damage is to the mitochondrial DNA, because any other damage to the mitochondria will get fixed.
Yeah, there's Leber's hereditary optic neuropathy, which is like an inherited damage to one of the DNA synthase genes. And it's like, or sorry, not DNA, ATP synthase genes that become proteins. And it's specifically like, it's like a single base pair or something.
Yeah, doesn't take a lot.
Yeah, and when
Doesn't
you
take a
get
lot.
that, like you eventually just go blind, I think in either one or both eyes, because your like eye cells no longer are able to produce enough ATP to
Yeah,
survive. So
yeah.
yeah.
There's no doubt that mitochondrial DNA errors, well, they call them errors, differences, variations, can be damaging to the function of the soma. And I argue that this is part of the change. The presence of senescent cells is one thing. So the more senescent cells you have, the more SASP you get, and the more that drives out interleukin 10. And the second one is the general inefficiency of mitochondria. And that a lot of that feeds through the lung genes. A lot of that feeds through, the acetyl-CoA level. And actually, I think a lot of that can be changed by exogenous citrate, for instance. Whether
The
triacetin helps is another question. It's a complex question. I've experimented a bit with triacetin, but it's quite hard to measure.
the association of so it's mitochondrial dysfunction leads to a downregulation of acetyl-CoA, and that's related to long genes because all genes need, acetyl-CoA, but the long genes need it more. That's what you're saying.
Yeah, because basically it's a question of to what, and this is where I don't know because I haven't managed to find anything in the literature, but we do know that when the histone deacetylases come around and take the acetyl group off the histone, that has an effect. Now, a longer gene is more likely to, what we find is the longer the genes, the more likely they are to not be produced. So you get alternate splices. So, prostate cancer is very much driven by alternate splices. Zygote's a really interesting thing to look at. The prostate's main function is to put exogenous citrate into seminal fluid so that the zygote, when it's actually initially dividing, is dividing in a sea of citrate. There may be a reason for that. Probably is a reason. I think I know the reason. I think you probably know.
I don't know. Well, can you tell me what is the reason?
Because the exogenous citrate goes into the cell, the zygote, and is used to produce more acetyl-CoA to make sure that transcription occurs.
Ah,
That's what
that's
I think is the case.
okay.
Well,
Yeah,
you have
that
to, evolution has said for mammals that zygotes need citrate.
mhm. Yeah, that's really interesting.
Yeah, it is interesting. Yeah. And why the prostate gland is mainly about inserting citrate into seminal fluid.
Yeah, I guess that would make sense because like presumably most of the mechanisms for the actual replication is, around the actual egg cells.
Well, the egg cells, obviously, they've got a set of nuclear DNA. So actually, I think this is where there's this complicated thing where sometimes you get multiple nucleuses. But without going into that, the fact is it needs to do a lot of transcription pretty quickly. It has to do it
Yeah.
reliably. For that, it needs acetylation. Right?
Okay.
And acetylation can only really be done with an acetyl group.
Right. Yeah, and I remember a while ago we were trying to figure out why like all the mitochondria that you get when you're born, like why there's never any issues with those. And I guess there isn't literally
Because you're
never any
born.
issues.
because you're born.
Yeah,
If there
exactly. It
was
is.
an issue, if there was enough of an issue with the mitochondria, wouldn't be born.
Yeah,
But you wouldn't even get to the stage of a miscarriage because nobody would notice. The zygote would die at such an early stage.
not with a bang, but with a whimper.
Well, not even a whimper or a bang. It would just be maybe a missed period, maybe not a missed period. It's sort of like, if the mitochondrial quality is too low, nothing much happens. And obviously, this question about fertility, and egg quality. There are it's not just about eggs, but eggs make a big difference.
Yeah.
Yeah, just so I have to, we're the programmed aging people, so I had to bring up programmed aging a little bit. Your theory is really about the mechanism of aging. I like it. I like that we like the mitochondria. We like theories that include that. It's often it's not emphasized enough. This relationship between the long genes and aging, that's also highly controversial, but I support that. Nothing you're saying is against programmed aging.
No, well, what it is, it doesn't really matter because what I'm concerned about is the mechanism. So I want to interfere because I'm older than you, and it's
But
useful to me to be more functional. So
if the body is destroying itself, then it's doing it on purpose. So just giving it more availability of the substrates not going to affect it. As you said,
well, there you go. That's where it comes down to experimental results, doesn't it? So basically,
yeah.
if at the end of the day, what I'm doing, which causes the various things that happen to me and the other people who are doing this, has a beneficial effect, then that's as far as I'm concerned. I don't care whether it's programmed aging or not, even though I don't agree with you that the body is trying to destroy itself. I agree with you that sometimes, this is the octopus example, sometimes the creatures have evolved to be self-destructive. That's true. And I agree with you that evolution will, to some extent, select against longevity. That is, they won't select to have creatures that kill themselves. But if there is a conflict between the survival of the young and the survival of the old, the evolution will select for the survival of the young. I agree with you about that. And I agree with you that you have examples where creatures have evolved to destroy themselves. I agree with you about that. But I don't think I don't see I see the aging as being a defect in design that doesn't matter to evolution.
Well, clearly evolution has the ability, let's say it is caused by a mitochondrial defect, just like it makes sure the babies are born young with good mitochondria, it has the ability to do that. And just like
Well, it
some,
doesn't matter to it because it doesn't want evolution selects for the success of the species, not for the success of the individual. Now, obviously, bowhead whales and turtles last a little bit longer, certain turtles. We as individuals from my point of view, I'm interested in health span, right? So I'm quite pleased that I have a mechanism to improve my health. So my kidneys function better, my heart functions better, blah, 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 blah. All of these different things. As far as I'm concerned, if I've managed to find a way of preventing diabetes, that's a positive thing. I'm quite pleased with that. Whether exactly, you see, at the end of the day, you should. I don't think you should think of evolution as being like a human being with motivation, sitting there rationally thinking from an ethical perspective, what shall I do here? It's a process which rewards reproductive success, right? And it will therefore reward reproductive success in favor of longevity of adults. But at the end of the day, what we need to do, if we want to improve people's health as they get older, and that matters more to me as I'm older.
well, why hasn't evolution selected for better maintenance of mitochondria?
Well, why should it? It
Because
already has.
it would make you live longer and have more
Yeah,
children.
Well, if that were to, what you have to think is something has to crop up. What you have is a system. We already have the system with the bottleneck for improving mitochondria, don't we?
That's for a newborn, but you
Oh,
could go
well,
on
yeah.
having more and more children, right?
We
Evolution should
also we
want that.
also have autophagy. So
Yes.
human beings last longer than mice.
Yes.
All right. So evolution has acted to make sure our Becklin ones function. Becklin one is a long gene, so you have a problem as you get older that Becklin one being a long gene is harder to get to function. So you don't get the autophagy at the same level. Now we have evolved to have systems to maintain mitochondrial quality, but these things aren't necessarily perfect. You shouldn't expect them to be perfect.
But why
What
not? It has the ability. Why shouldn't they be getting better and better and
what
better?
motivates it to be perfect? It doesn't,
Say,
we are in a world now where people live out more of their lives. Sadly, the world is getting more violent and there's more war. So there's a greater chance of people not actually managing to age because they die before they die from age. So,
but
I don't.
evolution, it's the incentive. It's clearly incentivized to select for more and more longevity. Do you agree with
No, I
that?
think evolution is incentivized to select for the reproduction of the species. That's what it's.
And more longevity would mean I have more and more children.
I'm not sure that's necessarily true, but we are where we are today, where actually the evolutionary selection is not really having much of a difference on a day-to-day basis.
And it might be, just to add, so David, it might be that, yeah, okay, you can have more children, but it might be more beneficial for evolution that your children have children. Thus, the sort of cutoff age at the age of 80.
Yeah.
After that, it's best to refresh the species from an evolutionary standpoint.
Yeah,
But that
but
would still be programmed aging, like the way I see it.
if um, we look at time scale, um, evolutionary timescales, particularly for creatures where you're having children like from the age of 20 onwards, are thousands of years. And we're discussing this online on a system that we would not have done 60 years ago. Right? Technology is moving on much more rapidly than evolution. Um, I don't think we have evolved to kill ourselves. But I do accept that evolution has selected against excessive longevity, where it might interfere with the reproduction of the species, because food availability is a key thing. When you look at how the bodies function to manage responding to energy availability, that's a key thing. But in the end, on a purely pragmatic basis, um, what do we want to do? Well, we'd like to not have diabetes, not have cancer, have functioning kidneys, etc., etc. And I don't mind what your views are on exactly what evolution is doing. I'm focused on trying to be healthy, except I drink a lot. But there you go.
That's okay. I drink
But
a little bit too.
one things I'm pleased about is that I have managed to find mechanisms so that I can handle alcohol far better than I used to when I was younger. Not by
So
reduced
I,
quantity.
I I think we're in agreement. You believe that evolution is selecting against what you call excessive longevity. I
Yeah.
agree with that too. For some reason, you're really against, it naturally extends. Okay, then it could possibly be selecting for self-destruction. And that's fine. You don't want to go there, and that's
Well,
that's
I
okay.
don't think it matters because I
Well,
don't
it
think it
matters
makes
for
a
this
difference.
question. Do you believe the body has a clock and
No,
it's so this down regulation of acetyl-CoA, you don't think it's there's no clock that triggered
I
it.
don't think there's a clock that triggers. I think it's a gradual process that you could calculate a formula worked out in various ways to see why it happens, why the Gompertz formula operates with the values that it has in it.
You know what
Because
would be fun? Maybe I'll send you stuff on like the substrate availability in the circadian rhythm, so it shows you that it's circadian regulated. That might convince you. I don't know
Well,
if
there's
I.
more. There's circadian, there's ultradian, there's annual, there's all sorts of cycles. And the circadian is a clear cycle. Ultradian is a very important
And the
cycle.
availability of these substrates that you think are important is
These go up
dictated.
and down. Yeah, no, all these things go up and down in those periods. But
So
that's
it's almost as if it's programmed by a clock.
no. The circadian is programmed mainly by blue light, but it will still run without
It's
the light. If you're
completely
blind and you don't have,
endogenous. It's
right,
entrained by light, but it's completely endogenous.
you're right. You are right about that. But I don't. I think I agree with you about that. I agree with you that there is essentially a clock type effect. And this is a question of what do you mean by a clock? Um, there is a process which, on average, will go at a certain frequency. Um, but I don't think it's, and there will, I'm sure you could, if you put enough effort in and could be bothered, calculate, work out a mechanism of linking autophagy efficiency to various other things. But Gompertz in itself only gives you a guide as to when people are going to die. And there's an interesting theory about Gompertz, which is that a collection of random failures. There is a paper on this. I can't find it straight away, but if you want me to, I will. A collection of random failures of components in a complex device result in a Gompertz style pattern of failure. But I don't know, but I don't I think saying it is a clock is over egging the pudding. But you see, the way the ultradian cycle, the HPA cycle works, which is about, on average, a 90-minute cycle, is driven by the interplay of various hormones. And is that a clock?
It's an oscillator. So by a
It's
clock,
an
I mean
oscillator. It's not, and it oscillates. It isn't reliably oscillating at the same frequency all the time.
it
Uh,
oscillates and
it oscillates.
it changes based on the signals it takes.
Yeah,
That's
it
exactly
oscillates.
right.
Now, puberty is a really interesting question. What drives puberty? Is it, for instance, an increase in heteroplasmy? There's a question. Well, here's what melatonin is considered to be an anti-puberty hormone. Melatonin acts particularly, even the pineal melatonin, trundles around the body and it acts to reduce reductive oxygen stress, particularly in the mitochondria. So it could be that melatonin particularly acts to reduce or to reduce the increase in heteroplasmy rather than reduce it per se, although it depends on autophagy and things like that. You know, what causes puberty has been happening earlier for people these days for some reason or other. Why? I don't know.
It's a big mystery. It's a big mystery what causes puberty. Yeah, it's something
Yeah, no,
that
I just say it's a really interesting question because it is, but puberty is part of development. Aging is part of development. Is it all actually driven by the same sort of thing?
I think there's a
Don't know.
clock, and I think everything in development, everything in embryogenesis, and everything in aging is driven by this clock.
Yeah, well,
Yeah,
I may agree with you. It's driven by something, but I wouldn't call it a clock. To me, I suppose I have a
even
degree
if
of
it's
physics. I think
yeah,
a clock should have some sort of accuracy to it.
well, it's taking signals and adjusting, so
Yeah,
it's a
well,
different type of clock.
it's a non-clock clock. All right, I agree with a non-clock
It's
clock.
a clock in the sense it's completely endogenous. You can remove all external signals and it'll tick. But you're right, it's constantly taking signals on the environment and
Yeah,
adjusting based on that. Yeah,
yeah,
so by clock, I mean an oscillator. It's keeping track of the number of
yeah,
oscillations, and I don't mind that it's taking external signals.
yeah. At the end of the day, I approach it from a pragmatic point of view, which is um, I'm interested in what can be done to improve health, particularly.
Yeah, and no, very good. I think I have a much better understanding of your theory now. Very interesting. But just to emphasize it, you know, I'm also just oriented. My main goal is to extend lifespan, health span, but the clock perspective says that that should be the target of the therapeutics. And any interventions that do not target the clock are going to have a
Yes.
negligible
Well,
effect.
okay. So I'm saying the mechanism is the mechanism of what you would call a clock, and I wouldn't really.
If this is the mechanism of the clock, yeah, then it could be successful. Yeah,
Yeah,
it would be
exactly. So
interesting.
the question is this the mechanism of aging, which you would describe as a clock, and I don't mind you describing it as a clock, but I wouldn't describe it as a clock myself.
Okay. Oh, if it's just, it's just semantics, then I'm
It's
on
just
board.
semantics, really. That's just semantics. I think there is a mechanism for aging. I think I have identified the key drivers in aging in what I've just spoken about.
The key
And if you want to
mechanism
call it a
again,
clock, I will say, okay, call it a clock. You want, okay, I'll drink to that.
heteroplasmy of mitochondria is the
And also,
mechanism of aging, and everything downstream of that.
it's a mechanism. It's the senescence is also a mechanism. So
So
it's a
that's also downstream of mitochondrial
no the senet
heteroplasmy.
well, it's a mixture of the two because senescence can be caused by external influences on a cell as well. Because but it's let's not. One of the difficulties is you've got these sort of two key things, senescence and mitochondrial problems. And they are both they go down the same route because they all hit the genome and the functioning of the genome. So you have the acetyl CoA side of it all, and also the ATP. Because I do think I saw some papers where there were lots of mRNA transcripts hanging around, waiting to be converted into proteins and not managing to get there. And there is clearly an issue with the RQC. There are papers on that.
I didn't realize the senescence was different in your theory.
Senescence is different because it's the IL-10 stuff. Senescence is patchy. So senescent cells cause further senescence.
It's patchy. That's what you said.
Yeah,
So,
senescence is patchy.
first of all, senescence is even very, hard to define. I remember maybe 2 weeks ago you were pressing someone on the definition of senescence. It's very hard to
It's
define.
very hard to define. But the senescence I'm talking about, which is stem cells getting stuck and not managing to differentiate properly.
Okay, so again, this is a very small number of cells that you can argue do
Well, I
this
don't know. It's
as
very small.
certainly the increase is very small, whatever your definition, or is
Yeah,
that
well, look, I don't know what proportion of senescent cells would fit into that category, but when you look at where do you find senescent cells? You find senescent cells in parts of the body which are rapidly dividing, which are the ones that are renewing. So Occam's razor would say, well, perhaps they're cells that have failed to renew.
So, John, you said that you don't think that there is a thing that's exactly a clock, but you also said that there kind of is. So, would you say that if you could intervene in the mitochondria somehow and restore them to some kind of like earlier state, would that then reset the entire rest of the organism to like
Well,
a more youthful state?
to the extent to which you can interfere in the mitochondria,
Hmm.
and to the extent to remembering the senescent cell, it's a two-pronged attack. It's not a single-pronged attack. And to the extent that you can actually remove the senescent cells, then you can get to a better state. But you can also intervene in the levels of acetyl CoA in the cytosol directly through an exogenous route. And that has an effect in itself in isolation.
Okay. So it's kind of like doing that is messing with the like to go with the clock analogy, that's kind of like messing with the hands of the clock to like reset it
Yeah, I
to
know
something
this
earlier.
is why I don't like the clock analogy, because you're messing with the function of the genome. What you're
Hmm.
doing is you've got the genome not functioning, and you want to make it work, so you make it work.
Okay.
You
I think
interfere with aspects of it, and it's particularly this issue to do with the acetyl CoA levels in the cytosol. And by interfering in that, you cause different things to happen.
so. If you could restore the levels of acetyl-CoA in this cytosol to
But it's
youthful,
dynamic anyway. The problem with these things is they're a dynamic equilibrium.
okay,
So,
but mhm.
and you've got to think this thing is continually going on. You've got your mitochondria trundling away. They're going around the Krebs cycle. They're generating protons, which are going on creating ATP. And every so often, some citrate pops out down the citrate carrier being swapped with malate in the cytosol.
Okay,
It's a
okay.
continual process.
But is there like a benchmark, like a youthful state that you could look at the levels of malates and citrate and such and say, like, this is what we're shooting for, like this is what we would
Well,
expect to see.
this is where it gets hard, doesn't it? Because you're trying to measure. You see, one of the things about the osteoporosis paper is they managed to distinguish between citrate, which was in the mitochondria, and acetyl-CoA in the mitochondria, and that which was outside the mitochondria in the cytosol. Measuring these things gets really hard. This is one of the reasons why it's been really hard trying to get to the bottom of what's going on. And I might be wrong. I'm not saying I'm the font of all wisdom. Um, but they're hard to work out because it's a dynamic equilibrium. So you're not sort of got something in a static state where you can measure it on one day, and that was where it was all the time. It's going up and down all the time.
Okay.
So it is hard. Yeah, these things to measure, like even citrate transport into cells, requires using alternative isotopes. So you get different. You mark citrate with isotopes, and you see where it's gone. But it's hard. It's not easy science. They're very small things, and you're measuring relatively small amounts. And it has been done. And there's a lot of good work being done, but it's not very easy. Not like watching a car road going round or something.
Okay. But so, do you think this is like a limitation on technology, or do you think that this is just something that's just cannot actually be measured?
Well, let's be straight. Technology has done very, very well. We can now measure heteroplasmy. That is, we can measure the extent to which in any group of cells or any one cell, there is a variation in the mitochondrial DNA. That's pretty good going. That's really impressive science. That's not a trivial task. It's been done very, very well. A lot of good work has been done by people over a number of years, which now enables us to measure something we weren't really aware of, say, 20 years ago. And we can measure it now. So that's good.
That's nice. But yeah, so like what I'm thinking is like if this is the key driver of aging, or I guess if it's like one half of the equation, the other half is stem cells, then if it's possible that we could identify what is the optimum state of acetyl-CoA and malates and whatnot, and then we could like induce that in somebody. Like
Yeah,
I
well, what I do is I experiment myself and my other people by eating a lot of it. Do you want me to go and get some so I can show you what it looks like?
okay, sure. Let's have a
Yeah.
live demonstration.
We'll have a presentation and then we have a visual demonstration.
Demonstration. That's right.
Yeah, it's a show and tell. No cats this time. I'm
No
sad.
cats. Well, I've shut the cat and dog out. If I was going to do a presentation, I thought having the cat and dog fighting would be harder. This is just some sour cherry juice. because it doesn't taste very nice. This is something I mixed earlier, which is a glass of citrate, 5 grams of citrate in here, right? Which is a bit cloudy, right? And so what I can do is I shall try and bring it on the camera so you can see the wonders of modern technology. All right, and this here is actually a mixture of citrate. All right, so in there you have a mixture of sodium citrate, potassium citrate, magnesium citrate, and calcium citrate, which I then put on a teaspoon in that cup, stir it, and then I'm just doing this around the corner, all right, because I'm putting a little bit of that's tart cherry juice in there, and then I can drink it. And that will then wander into my cells and cause an increase in the level of acetyl-CoA. And I get a bit of a tingling in my fingers in about 40 minutes. Partly it uses more vitamin B12 and vitamin B5, but I take more vitamin B12 and vitamin B5 at the start of the day to deal with that. And that has various effects, including I can show you if you want, I can use a different camera and show you my hair growing back on my head. Do you want me to use a different camera and show you that?
I think we can see it.
Wait, your hair is going back because you've cleared senescent cells. Is that
Yeah, well,
right?
because I've increased the acetyl-CoA level, and it partly clears senescent cells. It's a very slow process. If you go back to the well, it's you know, it's a gradual process. You see where I was at a number of. Let me just share the image that I started out with. Share screen. It's a very slow process, but as you can see, I have changed since 2019. Right?
Yeah, these pictures are funny. It doesn't even look like the same person.
Yeah, well, there you go. You see, this is a phenotype, isn't it? All right. Let me end that down then. Well, it is the same person. I know it is because it was me.
Wait, this is all 1 year apart. 19, 20,
Well, shall
21,
I go back?
22.
I'll go and give you that one again. We'll just do it. Let me do share screen again, so you've got that. Because in a sense, for all the waffling, that tells a story, doesn't it?
Wait. So this is when you started taking the citrate? Is that
Well,
what you're
no,
saying?
to be fair, the citrate started in early 2022. There were various other changes prior to that. I've been doing a
Uh,
lot of different things.
so 2019, you started just working out more, or
Well,
what'd you
not
do?
working out. No, I still don't do that much working out. I, I I don't mind talking about it, but it depends how much time you've got. But at the same time, yeah, look, what you have is there, you have me now, right? And you have me a few years ago.
Wait, on the center picture where you lost a lot of weight.
Yeah, that's right. That was 2020 to 2021. But
Oh,
you can
that's
see the
one
cat.
year.
That's the cat we had before.
Nice.
All right. So that's losing 45 kilos roughly. So it's roughly about a third of my body mass.
That's actually probably, one of the healthiest things you can do. It might have
Oh
more
no,
of effect than
I'm
the
not
citrate.
saying, you can argue about various things. And obviously I support the idea it was a good idea to not be obese anymore.
I see. So you've been on a health journey, and then your citrate is your latest.
Well, citrate and rapamycin and various other things as well. So you've got a very large number of interventions. And I obviously I'm heavily into measuring things. So, for instance, I measure the level of lithium in my blood serum, and I target to have a level of lithium that is orientated towards a particular biochemical process. So, molybdenum, for instance, I got some hair measurements on molybdenum. So you just do copper. People don't think about these things, and it's worth checking everything out because in practice, yes, your body will function with skewed up values because they will, but it will function better if you get things better. Like molybdenum is one I only recently started with molybdenum. I missed it for a long period of time. There are 4 key enzymes in the body that depend on molybdenum as an enzyme to exist. And they will exist in your body, but not in the amount. And they are key sort of detoxification enzyme. So you end up with a more toxic body if your molybdenum levels are a little bit low. So you want to just take them up a little bit, not a lot. You can obviously, a lot of these things you can overdose on. Pretty well everything you can overdose. But it's complex. Quantity. But it's sorry.
Yep, you can overdose anything basically.
Yeah, exactly. It's all a question of U-shaped curves and hormetic curves and all these sorts of things. So
the poison is in the dosage.
Exactly, it's the same thing that everyone always says. But from a biohacking point of view, measure it, know what's going on. I measure as I say, I do blood tests every week.
So I think I'm going to classify your theory. It's an accumulation of damage theory, and you've isolated 2 damage accumulants that you think are particularly important: the mitochondrial heteroplasmy and the senescent cells.
Yeah, it is an accumulation of damage, but the important thing is that it's the mechanism by which it prevents the cells from functioning properly.
And right? You think evolution perhaps had an incentive not to select for maintenance of these, but it's not actually has not selected for active processes of self-destruction.
That's true, except for in particular circumstances, because there are obviously circumstances, and you cited some examples where evolution clearly has selected for active destruction.
And the beneficial effects of like caloric restriction and exercise come from the fact that they somehow downregulate these 2 damage accumulants that you've found.
Particularly the mitochondrial one. It's not, and there's an interplay between the 2 because they all operate around the same pathway. As I said, Becklin-1 is a long gene, and Becklin-1 is the key gene that needs to be expressed as part of autophagy.
So if this theory is true, well, first of all, it means that during embryogenesis, that bottleneck that removes a large amount of the defective mitochondria, that is really what we need to study, and we need to utilize that in adults. That would be one implication.
Well, that would be interesting if you could do that. But you see, SOX2, for instance.
We can do it. We've done it. Everyone's done that at some point during embryogenesis.
But you see, if you take an autophagy inducer, you will get very tired because your ATP production will go down. Now, if you had something which was more extreme, the SOX2 type thing. So SOX2 induces autophagy as well. But the question is, can you survive it if you did that to all of your genes, all your cells at once? What would happen? Enhancing. I've got a cat now. If you want a cat, this is a different cat. It wants to be fed. You see, it's been hidden out of the way. It wants to be fed, so it's decided to try and chew my hair. I don't know why the cats like to chew my hair, but they do. But basically, you have to have mechanisms of improving health, rejuvenation, whatever you want to call it, that don't result in your death.
Yep.
Yeah, so it sounds like the way that you start your life with good mitochondria is by negative selection rather than
yes, I think in part, obviously, if your mitochondria are too bad, you don't start your life.
Yeah, like rather than there being a giant repair of all the mitochondria or replenishment, it's actually just getting rid of the possibility of it's like the great filter, exactly.
The bottleneck doesn't remove everything that's damaged. So your theory would predict that over geological timescales, organisms would be born older and older and older.
Oh no, no, that's the well. You could go through all of that, but the fact is that the selection is in part by organisms not being born, right? So if an organism is produced that is too defective because the mitochondria are too bad, it doesn't get born.
But some
Yeah.
organism is going to be born, and let's say
And some are,
almost
yeah.
all of your mitochondria is bad. Let's say if you're aged, and the bottleneck's not going to remove all of it. There's going to be some bad that's still
Well,
there.
this is something to look at in detail, right? I think the bottleneck shows what you would expect, which is a small number of really defective organisms being born, but a lot that are not defective, that are defective, just not being born.
Yeah, and I think that that's why that could be a reason why evolution selects against reproduction at an older age, because as you get older, the more chances are that your offspring would have damaged mitochondria.
I'm not
So,
sure. Yeah, I agree with you,
mhm.
but I wouldn't describe that necessarily as evolution selective. It's just it doesn't work so well because the mitochondria are not up to it.
Right, yeah, yeah. I'm just saying words. So yeah, you're probably
Yeah,
No,
right.
it's selected. There's menopause. They select. It's an active process where they can no longer have children.
Yeah,
well,
in humans at least.
that's also, I think, a long genes thing. I think it's a long genes thing as well. I had a look at the length of the genes on some of the sort of female proteins.
Wait,
And they're quite
menopause
long.
is associated with long genes. Is
Well,
that
no,
right?
I think again we talk about aging. The menopause itself, right? This isn't necessarily the quality of eggs issue. The quality of eggs issue, I think, is a mitochondrial issue. But the menopause itself is a long genes issue, I think, because it's an aging. I don't think the menopause is any
Hmm.
different to any other part of aging in its terms of it is aging. It's just aging happens at all ages, and
Oh,
some
you're
is more.
saying it's a somatic mitochondria heteroplasmy issue.
Yeah,
Hmm.
and obviously the senescence as well.
Oh,
Moving.
wait, so I know you went over this before, but the issue with long genes, I believe you said that it was just that they're more difficult to create because they're more energy intensive. So,
No, they take longer, so that the chances of the histone, basically this is this question of what exactly happens with the HDACs, but they, the I would expect it's because there's a greater chance that they haven't actually finished by the time the HDAC comes down and shuts it down.
oh, okay. Okay, so it's more difficult to make them, or the machinery of replication or translation is less effective, and so it just gets cut off by
Yeah,
the
that's right.
quality control thing.
Because it takes longer, the chances are at a higher probability that it'll get shut down.
Okay, I see. But so what does that have to do with menopause, though?
Long genes. I would argue that it's the longer proteins again that are not being produced.
And the long and the what am I trying to say? The longer proteins are needed to avoid
Well,
menopause.
yes, exactly. They're for the sort of the reproductive function to continue with longer genes. I
Okay.
had to look at some of the proteins at one point. It seemed to fit with the number of base pairs, which is like it's you're talking about, I think, about the 30,000 base pairs type territory.
Okay. Huh. Well, that's too bad.
Yeah, maybe talk about SAS. SAS is like these active signaling molecules that some
That's
people
right.
say.
That's for the well. It's obviously you've got interleukin 6, interleukin 10, and I think people have avoided interleukin 10 because it's anti-inflammatory, but I think it's that one that's causing the issue. But
Why
you
are
can see it with the
why are
Janus.
they doing that? Why are cells becoming entering a state in which they release these pro-inflammatory signaling molecules?
I think these particular cells are stem cells that have failed to differentiate, so they've got stuck. Right? They're stuck in the
That's
process.
fine, but why are they encouraging other stem cells to become senescent?
Well, I think the theory of it, from the body's point of view, is to actually, and I think one of the reasons why they put a mixture of this strange mixture of messages out of interleukin 6 and 11 and 10 is to not get the body to destroy them, whilst they hopefully get on with the process of turning into somatic cells. So I don't think it's I again, this is one of these things where there are different reasons cells become senescent. And I think this is a category. And I cannot say what proportion are of this nature. But what I can say is things like chronic kidney disease are associated with patches of senescence. A lot of the diseases of aging are very strongly associated with patches of senescence.
Well, senescence is associated with aging, so everything that's associated with aging is associated with senescence.
Yes, well, it's the word, isn't it? I suppose.
True.
Yeah. Are you saying that the SAS might be doing something good in addition?
The SASP, if you think about the SASP has pro-inflammatory and anti-inflammatory elements. And I think the reason it downregulates NF-kappa B is to stop cells in the area killing the cells off whilst they go through this particular stage of the cell cycle. Because what I think you've got to look at what NF-kappa B does in a cell. It enables a cell to produce ROS to attack cells nearby. Right? And that's used as a process of clearing up strangers locally. You know, it's sort of one reason why older people, in my view, older people get more warts, is they lose that ability to a certain extent by the levels of IL-10 floating.
They get more warts? Is that what you
Yeah,
said?
lumps and warts and all sorts of things. Yeah, I'm sure you must have seen that.
Yeah, everything. They get more everything bad.
Yeah, lots of bad things happen. Yes.
And so, okay, so we've identified two damage accumulants that are very important: senescence and
heteroplasmy.
Heteroplasmy. Let's say we can fix both these problems. How long would people live, in your opinion?
I can't say. I really can't. No way I can work that one out.
Well, shouldn't it just be forever?
I haven't got enough information to.
But do you think there's other damage accumulants that aren't as pressing, so it's not going to give us indefinite life? You think?
No, I think there are other. Well, obviously, nuclear DNA damage has an effect. I don't think I have enough information to be able to say how long that would give people. I would just say that would give you a healthier life.
All right, I'm going to show you some studies. I think it's going to rock your worldview. I'm going to accumulate them. You know, there are somatic cell nucleus transfer studies where they take the nucleus of an aged cell, put it, and they're fine. They live normal life courses, and they've done it for like 30 generations of mice. So whatever so-called damage occurred in the nucleus is completely irrelevant to aging, right? Yeah. And I think they've done the same thing for the mitochondria. Take the mitochondria in the old, put it in the young, and I think they're fine. So that would disprove this theory.
It doesn't disprove this theory at all. It proves this theory.
I take old mitochondria, I put it in an egg cell. I remove the mitochondria from the egg cell, put it there, and if it's fine, that means the so-called old mitochondria was not the problem.
The somatic nuclear one, I think, was the one I was referring to. If you've got the mitochondrial example, I'd like to read the paper.
Yeah, I'm going to have to look for. So the somatic nuclear transfer has done it.
Can I just ask, how much longer do you want to go on for?
You know, we usually end at like 2, but if you have to go, that's no problem.
No, what I'd do is I'd like a short comfort break, if that's all right.
That's okay.
Sure.
Yeah, so if I leave you talking whilst I disappear for a minute or two, yeah?
Okay, take your time. We have papers to read, yeah. And thank you for the presentation.
No, I'll just go and come back.
Okay. We should look for mitochondrial transfer papers.
Yeah. Yeah, like if the mitochondria are really the like culprits of all of this stuff, that would be pretty convenient.
Wouldn't it be nice if I could just replace the mitochondria? I mean, honestly, I think the clock might be the mitochondria. I mean, a lot of people, like Nick Lane's book, really emphasizes the fact that there's all this active machinery to destroy a lot of the mitochondria, including the paternal mitochondria. He emphasizes the paternal mitochondria. John was emphasizing the other mitochondria. It's an important point that people don't emphasize, and I don't know why, but it could be something to do. I forgot Nick Lane had that whole chapter on how it has to do with aging. Um, so
This is a vital question.
yeah, it's in all his books. It's in the other one too. What's the other one? So the cells are born young and they also destroy bad mitochondria. So that's why he thinks it's important. That's his argument. And I think that's what John was saying. And I think it could be important. The problem is the mitochondria is just so small that I guess that's why I thought the clock couldn't be there, right? It's just
Well, their
such
network is not small, and presumably the information that needs to be stored to maintain a clock is not a lot of information.
No, it might be a lot of information. I have to count every passage, every unit of time that has passed. I have to count. That means it's a lot of information.
True, but relative to the other stuff the body is keeping track of, maybe it's not
Yeah,
that much.
maybe it just doesn't need that many nucleotides.
Yeah.
Chondrial transfer agent. No one's done that experiment. We could easily do that in the lab, transfer mitochondria.
Are you saying nobody's done the experiment
We have to
of
find it. Mitochondrial transfer from mouse adipose-derived mesenchymal stem cell into aged mouse oocytes. Due to decline quantity and quality of oocytes related to age, the fertility of women over 35 years of age has declined sharply. Wait, they just say that as a fact? They know why? Oh, okay. Decline of quantity and quality. All right. We don't know why menopause happens. Okay. The molecular mechanisms that maintain oocyte quality remain unclear. Thus, it's difficult to increase the birth rate of women over 35 years old at present. Oocytes contain more mitochondria than any type of cell in the body, and any mitochondrial dysfunction can lead to reduced oocyte quality. In the 1990s, oocyte cytoplasmic transfer resulted in great success in human reproduction, but was accompanied by ethical controversies. What? I didn't even know they were doing this.
didn't know it was
Autologous,
from the 1990s, no.
yeah, autologous mitochondrial transplantation is expected to be a useful technique to increase the quality of oocytes that have decreased due to age. In the present study, we used adipose-derived stem cells from aged mice as a mitochondria donor. Adipose-derived stem cells from aged mice as a mitochondrial donor to increase the quality of oocytes in aged mice. Wait, they used aged mice as the donor to put in aged mice? I guess.
Yes. Because I guess they're looking at, can you de-age the mitochondria and then rescue aged
Yeah,
mice?
which you possibly could actually, yeah.
We
Yeah.
want a study that takes young mitochondria and put in old or old mitochondria and
Yeah.
young. That's what I'm looking for.
Yeah, no, that's what you need to find because I accept that it exists for the nucleus. That definitely does exist for the nucleus.
No, people do
I'm not
mitochondria
aware of its
transfer.
existence. Sorry?
They definitely do it, I think. I'm gonna have to look
Well,
more, but I'm
what I
pretty sure.
know is done for the purposes of fertility is for human beings is that there is a mitochondrial transplant done for mitochondrial DNA. Now that I know that's done,
Oh,
Say it
like
again.
um,
for
What's done?
a mitochondrial transplant, which is the three parent issue. That's definitely done
Yeah.
in
And I
small
guess
numbers.
that actually helps your theory if they
No, it
put.
does help my theory. I'm sorry, but yeah, I accept the nuclear one. It's not nucleotide. It's definitely. I don't think. I'm not aware of anything that challenges. Very happy if you can find something. We'll have a good read of it. But because I want to know what's right. If I'm
They
wrong,
take young mitochondria and put it in the old egg cells, and that is a fertility treatment, right? That's
yeah,
what you're
exactly.
saying.
That is yes, yes.
Oh, I see. And that does help your theory. But what we need is a study that takes old
Old mitochondria. Yeah,
mitochondria and put
exactly.
in young.
Yeah, no, but
Yeah,
I
has
think
it's an easy thing. Like people that have labs that transfer mitochondria could do this. Has no one done this?
I don't know. I'm not aware of it, and but again, there is a lot of literature out there.
I love these acronyms they come up with.
They love their acronyms.
Yes,
Hmm.
not surprising.
Should the U be lowercase in augment as well as the N?
Yes, I think you're right. However, let's not worry.
I had a lab meeting once where they were like, why I changed the acronym to my software. They got really upset. I said, hey, shouldn't we be worried about if I change the how it actually works rather than the acronym?
Yeah.
They're asking the real questions.
Is this what we want? To mitochondria. 20 minutes where we discuss hypotheses that suggest alternative treatment options using autologous mitochondria to potentially augment pregnancy potential in ART. Autologous transfer of mitochondria from patient's own germline has attracted much more attention as a possible new treatment to revitalize deficient oocytes. No, actually, it's not what we want. I need old mitochondrial transfer aged
I
into youth.
there yeah. There is a difficulty with it, of course, which is you've got a cell will have an potentially 1,000 mitochondria in it, which will have different DNA, and you've got to pick which ones you're going to identify and insert them. There is a the EVs do automatically do already sort of transfer mitochondria around the body, but
I guess we might have to do this experiment. Look at this migration of mitochondrial DNA into the nuclear genome in colorectal adenocarcinoma. That's not possible. They have different machineries.
well, it can obviously it's moved around, but the advantage of having the electron transport chain mitochondria in the mitochondria itself is the speed of repair. But obviously, it could be somewhere else. It doesn't have to be.
Yeah, it's different for different vertebrates too. Not
Yes,
it's
it is. It's
the
different. These things obviously vary. That's why we have species.
it's not clear why some vertebrates have this mitochondrial DNA and the other, as you said, most of the mitochondrial proteins are actually in the nuclear DNA.
Yeah, yeah.
And
But
then
the
other
ones,
vertebrates have different ones
yeah,
in the
it's
mitochondria.
the electron transport chain proteins particularly. Because if you think about it,
But
you
is
want.
that true just for humans, I think?
I don't know, to be honest. I haven't got enough knowledge there.
Maybe Stefan, ask ChatGPT if he knows of any papers. We need old mitochondria into young organism.
It's really hard, that really hard. You've got to somehow separate out the
Just
mitochondria.
take all the mitochondria. They do that. They can take just like they do and they put the young in the old. They do that for fertility treatments. Just do the same method, old into young.
So today I had some problems with my
I,
ChatGPT. Yeah.
I don't think anyone would necessarily want to take dysfunctional mitochondria and put them into a functioning cell in the hope they may suddenly function.
Well, to prove that it is
Well, yeah, I know.
dysfunctional.
Well, yeah, I just sort of.
Well, they wouldn't do it in
The thing
humans.
about DNAs
Yeah.
is you're taking the DNA. Your DNA. Well, yeah, it's. My cats are getting quite insistent. Would you mind if I went and fed the cats because they're getting quite stressed?
No, yeah, that's okay.
All right, come on, let's feed you two.
You're playing Go.
Yeah. Sorry, I got all my windows open. Transfer mitochondria from old to young. Jackson, try to find a paper too. It would be cool if we get a paper while John's here.
Spooky. Is my typing annoying?
I don't hear it.
Okay, that's good.
Ah, damn, I guess we're gonna have to do a lab. We're gonna have to contact one of these labs that do the transfer from young to old. See if they ever did it.
So we want to transform mitochondria from one cell to another. Or
Yeah, from an old cell to a young cell and to see what happens. We'd need a paper that did that.
is there a specific reason to emphasize from old to young, or just
If
for,
it
for the
lives a
tech?
normal if it lives a normal life course with the old mitochondria, then it disproves the theory that mitochondrial heteroplasmy is a key cause of aging.
Yeah.
Yeah, I only found papers that did the other way.
I found a review paper about mitochondria dysfunction in general. I'm just skimming through it right now to see if this is something they mention.
All right, this is what I'm gonna do this week. I'm gonna contact some of these labs. Like this lab did the other way. To evaluate the effect of mitochondrial transfer on embryonic development,
Yeah.
mitochondria concentrates collected from urine hepatocytes and fertilized murine zygotes from young and old mice. Nice.
Right.
In the 2PN stage were subjected to mitochondrial transfer. Oh, but from where? Into in vitro to evaluate embryonic development. Oh no, so they're all going. Wait. Is this what we want? Say, so they took mitochondria from old mice, and they looks like they injected it into something. Results: after extended in vitro culture, 37.65% and 20.91% of embryos from the young mice developed to blastocyte stage in the injected and control groups respectively, which is statistically significant. There was no difference in terms of hatching rates. Zygotes from the older mice that received mitochondrial transfer also had a better developmental outcome
You'll
than the
need to get the paper itself there.
Yeah, I do not know what they did. Well, they weren't trying to answer our question. I don't even know what question they are answering. Our results for the murine model provide direct scientific evidence that mitochondrial transfer improves embryonic development. However, potential risks such as mitochondrial heteroplasmy, nuclear mitochondrial interaction, and epigenetic aspects all deserve further evaluation before mitochondrial transfer is applied clinically. With advances of the assisted reproduction techniques, successful pregnancy rates have increased nearly 40 to 50% in some centers. However, there are still some patients who suffer repeatedly from failed trials, especially with poor ovarian reserves and poor embryonic development. There have been various attempts to improve results, including alterations of stimulation regimens, ICSI, co-culture, alternative culture methods, assisted hatching, and fragment removal, etc., but the results are still disappointing. It has been suggested that the introduction of a small amount of ooplasm from a healthy donor oocyte might rescue the function of oocytes that may have certain ooplasm deficiency. Ooplasm transfer has been attempted in humans, and a few pregnancies have been resulted. However, it is not yet fully clear how ooplasm transfer works. In recent years, the adenosine triphosphate producing capacity of germline cells has been correlated with reproductive performance. Perez et al. Microinjected a small number of mitochondria into the mouse oocytes and found that the procedure prevented oocytes from undergoing apoptosis. Yes, some of the oocytes killed themselves. Oh, interesting. But they injected a small number of mitochondria from ones that are producing a lot of ATP, I guess, and it prevented apoptosis. Their data provides a direct correlation between ATP producing capacity and reproductive performance and helped, at least partly, in elucidating the ooplasmic factors. In the same report, they also showed the occurrence of a common mitochondrial DNA deletion in unfertilized human oocytes are more frequent as compared with fertilized oocytes. Okay, so they took some common mitochondrial DNA deletion. That's fine. This indicates the status of mitochondrial DNA integrity is associated with potential fertility of oocytes. There is some very little evidence. Later, the concept of mitochondrial transfers was proposed in practice of women who suffered from repeated pregnancy failure and resulted in clinical pregnancies. Okay. Yeah, that is cool. However, there is still no proper preclinical evaluation about the efficiency of mitochondrial transfer on embryonic development. This also limits its application. To further evaluate the effects of mitochondrial transfer on embryonic development, we injected the mitochondria extracted from mice hepatocytes, so old mice, into zygotes from young or older mice to compare the developmental outcomes of zygotes with or without mitochondrial transfer. So they took mitochondria from old mice. Is that what I'm
Well, we don't
reading?
know how old the mice are in what mice sense, you know.
Okay, but they're older than
Well, let's have a
zygotes.
look down. Well, for
Yeah, preparation of fertilized mice oocytes. 4 to 6 week old young female ICR mice and older mice consisted of female ICR mice older than 20 weeks received, these are the ones that got the mitochondria.
yeah,
Intraperitoneal injection of 10 IU
it doesn't say where the
PMSG.
mitochondria is the next set para, and it doesn't say where they're from.
The mitochondria were prepared by a modified method described
Oh, I
by
have to look at
Pinkert
eight.
et al. Briefly, the mouse liver tissues measured as 2 times 2 times 1 millimeters were minced. Oh yeah, it doesn't say where they're from.
No, exactly. You have to read eight.
Wait, they have to tell us where they're from. Maybe the methods. Oh, no. It doesn't tell us.
If you try eight, you might get. Yeah, it's sort of the the problem with the mitochondria is you need to know which mitochondria you're transferring because you need, if you're transferring quality mitochondria, it's going to be improve the situation. If you're not transferring quality mitochondria, it's going to make it worse.
I see what you're saying. Like in an old mouse, let's say you happen to just choose the mitochondria that was really good.
Then if you get the better mitochondria, well, this is what I mean, my approach in terms of improving health is to improve the quality of mitochondria, which is to replicate the ones that are good and get rid of the ones that are bad.
Yeah, maybe just
And
them selecting it, and maybe the bad one died in a tube or something. So
well, you don't know. It's sort of I would say yes, it's a useful experiment to do in one sense, except if we know that the mitochondrial DNA is damaged and the mitochondria, I think it was this paper was saying
okay, but
mitochondria which
if
produce.
we assume that this is taken from old mice and it's their mitochondria is representative, it's not particularly good
Yeah, if it's
because
representative, it's a different thing. But it's the thing is what I think this is the paper that said we're looking at the mitochondria which produce more ATP. Well, those are the better mitochondria. The whole point about this is to have mitochondria that are efficient in production of ATP, which should also mean they produce more citrate, and therefore you get more
yeah,
acetic
I
acid.
mean that's a fair criticism. They weren't trying to answer our question.
No, I know. I don't. I'm not sure anyone has tried to do that.
But in this
I think
paper, it looks like they took mitochondria from over 20-week mice and put it into the oocyte, and it lives a normal life course.
well, this is the point about the oocyte is that there is this um, bottleneck, the sort of DNA bottleneck that
Yeah,
occurs at
did
some
they do
point.
it before or after the bottleneck? That's
I
also
don't
another
know.
good
I don't
question.
know. It's a question that involves, unless they say whether it's before or after the bottleneck.
With
You then have to
transfer
find out when the
procedure,
bottleneck is.
the holding and transfer pipettes were mounted on the
Yeah.
microscopes up to 5 zygotes at
2 p.m.
the
stage.
two.
Well, is that before or after it? Do you know?
I have no idea. Stage were placed in the dish, one in each herpes droplet. The dish was placed on a heated stage of the microscope, and the droplet of the mitochondrial suspension was visualized. The transfer pipette was lowered into the droplet. Yeah, actually, so we have to look up TPN stage.
Yeah, 2PN score is generated from the grading of pronuclear size and alignment, nuclearly and alignment and distribution, and cytoplasmic halo. 2PN stage, mitochondria.
Wait, so what am I looking for? So I'm looking for they took it old mitochondria, and how are we gonna just get a representative sample? I guess we'll just have to take a bunch of samples. And we inject the mitochondria, and we're looking for a study that does it after the bottleneck. Is that
Yeah.
right? After the bottleneck. Is the bottleneck the official term for that?
Yes.
Mitochondrial matters. Mitochondrial bottlenecks, self-assembling structures, and entrapment in the female germline. Mitochondrial replacement therapy, a procedure to generate embryos with nuclear genome of a donor mother and the healthy mitochondria of a recipient egg, have recently emerged as a promising strategy to prevent transmission of devastating mitochondrial DNA diseases and infertility. The procedure may produce an embryo that is free of diseased mitochondria. A recent study addresses important fundamental questions about the mechanisms underlying the maternal inheritance and translational questions regarding the transgenerational effectiveness of this promising therapeutic strategy. This review considers recent advances in our understanding of maternal inheritance of mitochondria, implications for fertility and mitochondrial disease, and potential roles for the Biani body, an ancient oocyte structure. In mitochondrial selection in oocytes with emphasis on therapies to remedy mitochondrial disorders. Monoeukaryotic cells co-evolved in a symbiotic relationship that began between microorganisms, archaeobacteria, and ancestral mitochondria 2 billion years ago. Along the way, much of the original mitochondrial genome was lost, with just over a few dozen genes among more than 1,000 retained in the mitochondrial DNA. The mitochondrial genome of humans is comprised of 37 genes that encode the transfer RNAs and ribosomal RNA required to translate the proteins of an electron transport chain, as well as proteins that are necessary for oxidative phosphorylation. The other gene functions that are required by mitochondria are supplied by the nuclear genome of the cell. The mitochondrial genome is organized into nucleoids composed of single or multiple copies of mitochondrial DNA due to fusion and fission cycles that ensure each daughter cell receives highly functional mitochondria during mitotic divisions. Yeah, actually, it's completely independent of the cell's mitotic division, I believe. So that's misstated. These fusion and fission cycles can also generate mitochondria with distinct morphologies and even interconnected networks that with presumably unique functions. Unlike the nuclear genome, the mitochondrial genome is not transmitted by sexual reproduction, but instead, in most animals, mitochondria are strictly inherited from one parent, usually the mother. Either before or after fertilization, mixing of maternal and paternal mitochondria is averted by elimination of any contaminating paternal mitochondria by seemingly diverse mechanisms, including ubiquitination modification and proteasomal degradation in mitophagy. The involvement of conserved components, e.g., autophagy machinery, and the conserved reliance on redundant mechanisms to eliminate paternal mitochondria underscores the evolutionary importance of uniparental transmission of mitochondria, but the rationale behind this investment remains mysterious. Oh yeah, does the uniparental inheritance fit into your theory at all, John? Why is does the fact that the paternal mitochondria is almost always among the mitochondria that's destroyed, does that fit into your
it doesn't make, I don't think it makes any difference to my idea. Because my ideas are basically around the fact that it is the status of the mitochondria is driven by the mitochondrial DNA, and
it doesn't matter whether it's a young father or an old father, it's almost always destroyed, right?
Well, the father's stuff tends to be destroyed, and that may be an argument about why it doesn't really matter when my oldest child was born when I was let's think 52.
But your theory is that the good mitochondria or the bad mitochondria is preserved and the bad mitochondria is destroyed, but you can get a very young father and still destroyed.
My theory is, well, this is a bottleneck, is that basically at the creation of the zygote, at some point, the variation in mitochondria is reduced. If you then end up,
and the paternal is such a small amount, it's always going to be viewed as an outlier. I kind of see what you're saying.
The variation is reduced. After that point, if the zygote's mitochondria is not good enough quality, then the creature's not born. All right, so you just don't get a birth. You don't get a failure after birth. You do actually get some failure.
So again, when the sperm comes in contact with the egg, the sperm does have some mitochondria, and then
and that gets destroyed. I don't know.
There's a bottleneck, and a bunch of things get destroyed. And you're right, almost always the paternal gets destroyed.
Yeah.
Yeah, I'm just trying to see how that. Yeah, it works in well with your theory, I guess. Despite the single parent of origin, variations in mitochondrial DNA between a mother and her progeny and between siblings exceeding levels expected in a single generation have been observed.
Yeah.
Is that true?
Yes.
Oh, because of the bottleneck. This variation can be extensive and has been observed in Holstein cows, mouse models, and patients. For example, a heteroplasmic mother, an individual with more than one type of mitochondria due to accumulations of mutations, can produce progeny with only the mutant mitochondrial DNA homoplasmic. Such enrichment of mutant mitochondria indicates the existence of a mitochondrial bottleneck. Yeah, wait, so heteroplasmic mothers have children of normal lifespans. Bottlenecks enforce mechanisms that omit cells or individuals. In this context, mitochondria with extensive mutational burden in mitochondrial DNA, ideally such that only the healthiest mitochondria are transmitted. In all animals, mitochondria and other organelles are asymmetrically enriched within oocytes. In animals that use maternal inheritance of germ plasm, the substance that contains determinants of the germline of the next generation to specify the germline, mitochondria are enriched in areas where the germ plasm, the components that will specify the germ cells of the next generation, localized and reviewed in, that was a bad sentence. Thus, subcellular enrichment of mitochondria has led to models whereby the oocyte selects the healthiest mitochondria for the next generation by mechanisms that remain to be understood. Mitochondrial bottlenecks are thought to occur in the developing germline, both during oogenesis and later in embryonic primordial germ cells. Oh, I didn't know it happens multiple times. Several models of mitochondrial inheritance have been proposed and include random segregation or stochastic mechanisms, as well as more direct selection mechanisms that will be discussed briefly herein. Yes, certainly not stochastic. Okay,
Well,
yeah,
it can be stochastic simply because basically, if you limit the variation and the creatures which have the worst mitochondria die off, then that selects effectively for creatures with a limited range of mitochondria that are of good quality. But it also,
so it just randomly selects, and the ones that are good
no,
survive.
I said it could be. I think
yeah.
it
I mean, it
says
always
likely to be
gets
something based on membrane potential, but because membrane potential is the easiest way of distinguishing. It's what they use, what cells use for autophagy.
okay. I take it back. Could be stochastic,
It
but it
could
always
be
gets.
stochastic. I'm not saying it is stochastic. I'm just saying it could be stochastic.
All right, that's fair. Heteroplasmy, yeah, they said nothing to disagree with your theory yet. Heteroplasmy, mitochondrial segregation, and disease. In an individual with a mitochondrial disease, different cells or populations may have differing amounts of mutant mitochondria, ranging from the complete homoplasmy, individuals with all mutant mitochondrial DNA, as occurring in Leys syndrome,
Yeah.
to more tissue-specific accumulation and manifestations, such as the blindness that occurs in patients with Leber hereditary optic neuropathy. Leber neuropathy. Organs and systems with high bioenergetic demand, such as the brain and heart, are most sensitive to mitochondria-associated pathologies. Stochastic and tissue-specific selection of mitochondria are mechanisms that have been proposed to explain variation and accumulation of mutant mitochondria and mitochondrial disease in somatic cells. Unlike nuclear DNA, mitochondrial DNA replication is not limited by the cell cycle. Consequently, according to the stochastic or mitotic segregation mechanism, a heteroplasmic cell can generate progeny with distinct mitochondrial DNA composition over a short time, depending on the extent to which mitochondrial DNA replication occurs before or after the cell divides. According to this model, most cells in a heteroplasmic individuals would be expected to have a random distribution of normal and potentially pathogenic mitochondria. Such heteroplasmic individuals are healthy if the cells have sufficient number of activity of normal mitochondria to provide adequate respiratory or other functions. In contrast, cells with an unfavorable ratio of normal to mutant mitochondria show signs of disease. This stochastic pattern of inheritance can explain why mitochondrial mutations do not affect all cells in an individual equally. An alternative possibility based on examination of heteroplasmic mice indicates that variation from cell to cell in a heteroplasmic individual may not simply be stochastic, but is influenced by tissue-specific mechanisms or bottlenecks that drive mitochondria selections. Why is mitochondria homogeneity beneficial? Let me skip some of this. I want to know when the bottleneck happens. I don't know if they're going to talk about that.
Yeah, I haven't really found that. It's one of these things where you might be better searching for more recent papers.
Yeah, actually, they might just not talk about it in this paper. Maybe I'll say, or actually, they did talk about it, but they did cite some papers. Yeah, 4 through 11. Yeah, maybe we'll check the most recent. Yeah, molecular basis for maternal inheritance. That could be good. Uniparental inheritance of mitochondrial DNA is an evolutionary trait found in nearly all eukaryotes and many species, including humans. The sperm mitochondria are introduced to the oocyte during fertilization. The mechanisms hypothesized to prevent maternal mitochondrial DNA transmission include ubiquination of the sperm mitochondria and mitophagy. However, the causative mechanism of paternal mitochondrial DNA elimination have not been identified. So maybe there's not just one bottleneck. Maybe it's just a continuous process of destroying certain DNA through various mechanisms, or people just don't know.
I'm not sure it's necessarily that known. You know, it's only recently that been able to measure heteroplasmy. I don't know exactly when that started, but it's not like it's the last 10 years.
It's still debated, you know.
I don't think heteroplasmy, the existence of heteroplasmy is debated. I just think it's harder to measure what's going on. And then you have, when it comes to autophagy and mitophagy, you have this issue that the cell can pick up mitochondria that should be got rid of by virtue of problems with their membrane potential. But if they haven't got a problem with the membrane potential, but are still not working properly, like problems with ATP synthase, then they're not got rid of. So that itself causes the problem.
We found that mitochondria in human spermatozoa are devoid of intact mitochondrial DNA. Well, we found that mitochondria in
Well, that would
humans.
explain why they haven't gotten none of it follows through, wouldn't it?
Wait. Well, it doesn't make sense. We found that mitochondria in humans, so spermatozoa,
Yeah,
so it
but
means
they might have the mitochondria, but not the DNA for the mitochondria.
Oh, I see.
You
And
see what I mean? So it's sort of like
yeah.
you know, I had read up on these things to a certain amount, but I wouldn't claim to have read up on it fully.
What happened to their DNA then? And
No,
lack mitochondrial transcription factor A, TFAM, the major nucleoid protein required to protect, maintain, and transcribe mitochondrial DNA. I've never heard that the sperm just doesn't have mitochondrial DNA. During spermatogenesis, sperm cells express an isoform of TFAM, which retains the mitochondrial presence ordinarily removed upon mitochondrial import. Phosphorylation of this pre-sequence prevents mitochondrial imports and directs TFAM to the spermatozoan nucleus. TFAM relocalization from the mitochondria of spermatogonia to the spermatozoa nucleus directly correlates with the elimination of mitochondrial DNA, thereby explaining maternal inheritance in this species.
Which
Okay,
species? Is that human beings or somebody else?
human, yeah.
Well, quite possible. Yeah.
I'm going to read more about it. Well, I'm going to contact the other study's authors. So I'm going to ask them specifically about this idea of taking old mitochondria. We'll read a little more of this. We'll go to 2:30. That's okay.
Yeah, I'm all right. No,
All right. Uniparental inheritance of mitochondrial DNA is one of evolution's few enduring pillars. Except for some fungi and mussels, most eukaryotes inherit the mitochondrial DNA from a single parent, most commonly a mother. The uniparental mitochondrial DNA inheritance pattern evolved repeatedly, and a multitude of mechanisms have developed to exclude the paternal sperm mitochondria from a zygote. This suggests the existence of selection pressure, as retaining the uniparental inheritance appears to be positively beneficial. However, the benefits of uniparental inheritance for fitness are not immediately clear. Intuitively, having genetically divergent copies of mitochondrial DNA may complicate their cooperation with the nuclear genome and generate selfish mitochondrial DNA molecules, which replicate efficiently but cannot support adequate energy production. Yeah, John, you would be interested. I don't know if you've read any of Nick Lane's books. He's a professor, and he does mitochondrial. He has a mitochondrial theory of aging, actually maybe similar to your theory of aging. He's a very good writer. He was the one that got us into this mitochondria. We were into it for a while. He's all about, not about mutations of the mitochondria. He's all about the mitochondria having to work well with the nuclear DNA. I actually forget exactly what his theory is, but we did talk about it. I feel bad. I don't know if anyone remembers. Jackson, do you remember?
If you hunt up the right book to read through, we could have another session to look at that.
Yeah. I'll reread it. He talks about it in his book, The Vital Question, the last chapter. I'll reread it again. I'll be more prepared.
I'll see if I can find it at the library. Because, yeah, I wasn't actually able to read that one because I never had it.
We had another Thanos was another. He's a regular here, even though he's not here today. He was really into that book.
Yeah. He's like here every 2 sessions that we have. Like he'll skip 2 and then be here for 1.
Uh-huh. Indeed, heteroplasmy, of the presence of different mitochondrial DNA haplotypes, results in genetic instability and adverse physiological effects, behavioral and cognitive complications in mice. Despite the overwhelming dominance of uniparental inheritance, The underlying molecular mechanism depends by parental inheritance is not understood. Profound downregulation of mitochondrial DNA levels during spermatogenesis occurs in some non-human species, reaching 0 in one report in mice. It has been reported that ubiquination of the sperm mitochondria leads to their targeted degradation by the proteasome-descendant proteolytic machinery after fertilization in mammals. Selective degradation of the paternal mitochondria by autophagy, a process known as mitophagy, in the embryonic cytoplasm has been documented in nematodes. However, experiments show a lack of mitophagy in mammalian oocytes. Is that right? And thus the mechanisms of maternal inheritance of human mitochondrial DNA remain obscure. Previous studies reported large variation in mitochondrial DNA copy number in the human spermatozoa from 1 to
When is this
1,000.
paper? What date is this particular paper? Is it 23?
Yes.
Yeah.
Nature Genetics.
It's pretty well as current as we're going to get.
Yeah. I don't recognize these people, but it's a prestigious journal.
I'm not being critical of it. I'm just saying this is something which I would think is rapidly moving.
Yeah. No, I get it. We want to
the
get
technology has only recently enabled tracking it.
To accurately measure the mitochondrial DNA copy number in human sperm cells, we use droplet digital PCR, ddPCR, which allows a determination of the absolute number of DNA molecules in a cell. Because the method does not require isolation of the genomic material and allows quantification of nuclear and mitochondrial genes in the same sample with high analytical sensitivity and a limit of detection below a single genome copy per microliter of a sample, ddPCR is ideally suited for measuring low copy number nucleic acids. Amplification targeted two single copy genes in the nuclear genome, TBP and TEFM, and two mitochondrial genes, CYTB and ND1. We found that the sperm cells contained on average 0.58 copies of mitochondrial DNA. Analysis of the whole genome sequencing data of bulk DNA suggests that each spermatozoan contains on average 0.14 copies of mitochondrial DNA and consistent with ddPCR measurements. Wait.
The and is to do with the tables, though. But it's still.
Oh, I see. Yeah, sorry, I read it wrong. The copy number measurements have not been affected by mitochondrial DNA deletions as verified by deep sequencing of long-range PCR products of bulk spermatozoa DNA and ddPCR experiments using multiple primers annealing to the conserved regions of the mitochondrial genome. Each spermatozoan contains 50 to 70 mitochondria, corresponding to less than 0.01 mitochondrial DNA molecules per mitochondrion. So this paper is reporting that something is happening to the mitochondrial DNA of the mitochondria of the spermatozoans.
Yeah, say when the sperm is created, they're created without mitochondrial DNA. They have mitochondria, but without the DNA.
Very strange.
It goes wrong a little bit.
While extremely low, this number likely accounts for the background mitochondrial DNA detection in a few contaminating cells, namely leukocytes, which can harbor up to 100 mitochondrial DNA per cell in their mitochondria. Okay, but we actually want to read about the mechanisms of destroying not just the parental mitochondrial DNA, but all.
Yeah, the problem is you've got two separate mechanisms here. You want one mechanism that prevents the spermatozoan mitochondria from having mitochondrial DNA. And
yeah,
the
that's
second one
interesting.
is the bottleneck.
But this is different than the bottleneck. Yeah, I see what you're saying now.
No, it's all interesting stuff, but it's different to the bottleneck.
Yeah, I actually want a paper about the bottleneck, if you guys have it.
I once won about that too.
How come it's so hard? Can't I just type in mitochondrial bottleneck?
I think the problem is a lot of papers assume it exists because its outcome seems to appear without knowing the mechanism.
Yeah, I mean, it clearly exists, right? No one doubts that. Or, right? We're not doubting that. It's just I guess we don't know the mechanism.
Yeah, effectively, that's the problem.
I actually didn't understand that people didn't know the mechanism.
Well, as I said, this is an area where the technology has moved on a long way very recently. So it's hard to get the answers.
Naturally heteroplasmic clam. Clams don't age, right?
Clams?
Clams, they don't age, is that right? Or maybe just some clams?
No, actually, I'd never heard that clams don't age.
I think that's true. Might want to double check me.
Hmm. Some clams have life cycles of only 1 year, while at least one has aged to over 500 years.
Nice.
That's nice.
What is that species of clam?
Let's find out. Apparently, this individual clam's name is Ming.
Ming? Oh,
Yeah,
not the species name, the actual given name.
yeah, exactly. Let's see. Oh. It is in Arctica islandica. That's the name of the species.
Arctica islandica.
Yeah, Arctic island. Apparently, when the researchers opened it, it died, so it could have lived even longer.
Oh, are you serious? They killed a clam that's 500 years old?
Yeah,
That's
uh,
funny.
this is what the article says.
How do they know
Um,
it's 500 years?
yeah, that's a really good question. Um, let's see. Um, they counted the growth rings inside the clam's hinged ligament.
That's fair though, right? I think that's what they usually do. They grow like trees,
Yeah, I guess so.
and it's like seasonal, so they have different rings.
Yeah. Apparently, you can count the rings that are on the outside. You don't have to count the interior ones. That's what this article said.
Mm.
That freaking sucks.
Inheritance of mitochondrial genome of the mitochondrial genome does not follow the rules of conventional Mendelian genetics. The mitochondrial DNA is present in many copies per cell and is inherited through the maternal germline. In addition, mutations in the mitochondrial DNA will give rise to heteroplasmy, the coexistence of different mitochondrial DNA variants within a single cell, whose levels can vary considerably between cells, organs, and organisms. The inheritance and subsequent accumulation of deletion of deleterious variants are the cause of severe progressive mitochondrial disorders and play a role in many other conditions, including aging, cancer, and neurodegenerative disorders. Here we discuss the processes that give rise to cell-to-cell variability in mitochondrial DNA composition, focusing on somatic mitochondrial DNA segregation and on less conventional sources of a heteroplasmy, non-maternal inheritance of mitochondrial DNA combination, understanding how mitochondrial DNA variants and mutations emerge and evolve within an organism is of crucial importance to prevent and cure mitochondrial diseases and could potentially impact more common aging-associated conditions. So they seem to have a similar mitochondrial theory of aging.
Yeah, I think what they probably haven't got is the mechanism through acetyl-CoA.
Yeah.
They've linked mitochondrial failure, but not actually linked it through to through the citrate carrier. And
Are you the first one to emphasize acetyl-CoA?
well, obviously that paper on osteoporosis is where I got the idea from. So really, it's them. I'm the first person to go on about how it's everywhere and you can just chog citrate. All right. But it's that paper that's the key. To be fair to them, they I yes, okay, they used acetate, but I used citrate. But basically, it was that paper that set me on the path, because that basically what that does is it proves a mechanism for osteoporosis. And what I've done is generalize. I say, well, what if that mechanism is valid for all age-related diseases? What's the link through to senescence and all that? Why is it a reinforcing system? And then I've taken it on to, and why is my because I start with the assumption, which is a very simple assumption. We know a number of tools which mitigate aging. So any mechanism that is a mechanism which defines how aging happens has to be a mechanism which is affected by the tools that mitigate it. So if you don't have a complete answer to all the valid tools, and I don't include that and add boosters, then you've not got an answer. So I looked for an answer which was linked to all the things we know that affected, and this paper which gave a good. This gave when you look at it, there's loads of papers pointing in the same direction. But this one paper was very obvious on the role of the citrate carrier. Although, actually, this applies to senescence. And in fact, it's not necessarily a failure to replicate the citrate carrier that's the problem. Where you have a mitochondrion that is not functioning properly, because if you've got a mitochondrion that's not functioning properly, even if it's got enough citrate carrier, it will still not produce enough acetyl-CoA. So but I was very impressed by this paper. There's some other ones on the citrate. There's sort of the non-canonical citrate cycle. Was a vaguely helpful paper. This paper was really very good. This is such a good paper. I was very impressed.
Okay, I'm gonna read this paper more closely.
No, it's a very good paper. It's sort of, but it gives you essentially a complete argument as to how osteoporosis happens. So you have to ask yourself, well, take atherosclerosis, take sarcopenia. Both seem to have similar sorts of causes, which is the failure of cells to differentiate. And then you go on to chronic kidney disease. All these things are just it's all
Well, here they're saying the cells, your body naturally turns things over, but it breaks things down, but then it doesn't build it back up.
That's right, yeah. What it is, the body naturally renews, but the renewal process goes wrong.
Yeah, it's funny. I was telling you before that, like for the loss of muscle mass, your body actively destroys it and doesn't build it back up. That's true. And you can knock out the pathways that actively destroy itself and it stops doing that. You don't lose muscle mass.
Yeah, or alternatively, you can repair the pathways that enable it to recreate muscle mass.
Yeah.
So, for somebody like myself, I am stronger than I have been in the whole of my life. And I'm not a massive gym buff. I just I do a certain amount of exercise, a certain amount of resistance training, but not masses of it. I'm not really heavily into that.
Well, I want to see the most convincing argument from this paper. Restoring histone acetylation rescues aging-driven osteogenic defects. To
Yeah,
further explain the citrate carrier histone acetylation axis, we use 1,2,3 benzene tricarboxylic acid, BTA, a specific citrate carrier inhibitor 33, to modulate citrate carrier activity in young mesenchymal stem cells and analyze levels of nuclear mitochondrial acetylysine signal. Inhibition of CIC activity in young cells decreased levels of nuclear acetylation and redistribution of the acetylysine signal inside the mitochondria. Extended data, figure 5BD, mimicking the results observed in age mesenchymal stem cells. In a complementary approach, we added sodium acetate directly to the medium of age cells. Oh, they added sodium acetate, kind of
yeah.
similar to
Well,
what you're
well, it's the
doing.
same sort of thing, isn't it? I use
Yeah.
citrate, I use acetate.
Why did you choose something different?
Um, because it's a citrate carrier. I have used acetate as well. I'm using acetate at the moment because I use acetate through the ethanol to acetaldehyde to acetate routine. Um, but I just thought citrate just seemed a better way of going about it. One of the problems with ACCS2 is that when you have higher levels of acetylation, that particular enzyme is inhibited. And I think it happens a bit with ACLY as well. So it's not perfect.
Sodium acetate is an exogenous source of acetyl-CoA and can be converted into acetyl-CoA in the cytoplasm by the ACS enzyme, whose
Yeah,
levels
exactly.
did not change with age. Impressively, supplementation of age cells with sodium acetate rescued the loss of histone acetylation, resulting in levels similar to those of young cell, figures 4AB. Collectively, these data corroborate our observation that loss of the citric carrier upon aging is responsible for the reduction of histone acetylation levels, providing a mechanistic link between mitochondrial metabolism and histone acetylation upon aging. Figure 4. Sodium acetate remodels chromatin and improves osteogenic capacity in age mesenchymal stem cells. So, what are we looking at? Young control, old control, and here's the old cells with acetate, and they stained it for H3 acetylation in green. And are they trying to say that the old looks more like the young? All right, acetate did something. I don't know if the old looks more like the lung. All right,
Yeah, acetate is a acetate can work, but I think citrate's better.
and so here's
Yeah.
H3 staining to show, I guess they want to show you that this acetylation is matching up with H3. And here's the DAPI staining, which does the nucleus. So these are things are in the nucleus. Okay. H3 acetylation, H3 MFI. What's MFI? Young control, old acetate. So old acetate looks more like young control. All right, maybe. Oh, PCA. This is what I do. I do hybrid matrices. All right, they took PCA of something and they got young, old, old. And so according to the PCA analysis, the old acetate, I guess PCA1 was the age factor and the old acetate was younger according to PCA1. Okay. RPKM, don't know what that is. What are we looking at here? Alizarin red S area. It looks like they have some figure to show that the cells are healthier. This figure is supposed to show that the cells are healthier. This is supposed to be a quantification of this figure. And these figures show that the mechanism by which they're healthier is restoring H3 acetylation. I don't know. I mean, I know all papers have like, this might be impressive to someone more familiar with what things are supposed to look like. But I don't know. I'm not taken aback. Oh, these are nice pictures. Yeah, that's what the mitochondria actually look like. This is a cool picture. What is this? Electron microscope? Gee. Represented images of MDVs highlighted with arrows in young and aged cells and high magnification of arrows in boxes. What were they? What type of images? Just representative images.
Yeah, those are definitely electron microscopes.
Yeah, these are so small. They must be, right?
Yeah, exactly. And that like color scheme, that's not a stains or light microscopy or something. That's definitely an electron.
Yeah, aren't the mitochondria? They are beautiful.
Yeah, they're neato.
All right, so old mitochondria with arrows are pointing to something. I'm not sure what they're pointing to. Old magnified. All right. Nuclei were stained with DAPI. Oh, no, that's the above image. Represent images after immunostaining of cells. Intensity plots represent image after immunostaining against citric carrier and quantification and BMFI of young control and young E65D treatment cells. Nuclei were stained with DAPI. So here is the staining. Old. And these were treated somehow. Yeah. John, do you know of any studies where they did a similar protocol that you're doing, but in not just cells, but in animals like mice and see if they live longer? Oh, we lost Jackson. Yeah, actually, I might head out as well, but yeah, it was great. Yeah, thank you, John, for the talk.
Right.
Thank you, everyone, for coming. Yeah, I'm gonna read more about the paper, and I'm gonna try to look and see if there's any evidence of a mitochondrial transfer from old to young to see the tested theory. But I think I know a lot more about your theory now. And yeah, thanks so much
Thank
for
you.
sharing with us.
All right. Well, if you want to shove it on YouTube, or I could do it on my account, then please do.
Okay. Yes, Stefan, you have the recording.
I
Maybe
will put it on the server and then you can access it from there. I will put the link on the Discord.
okay. Yeah, I
Lovely. Thank you
think
very much.
Yeah, it'd be great to let people know. All
All right.
right. Yeah. Thanks
Yeah.
everyone for coming, and thanks for the presentation.
Okay, bye-bye.
Bye.